Hello, how are you doing? Hola, ¿cómo están? What will be in German? I don't know in German. So I'm very bad with presentations, so sorry about this. We're going to start this symposium that we organized with many different funders, so a lot of help from many different people. So I have to acknowledge first the Office of Research from the university, Universidad Católica de Chile, that actually gave us some of the funding for the invitees that we have in the symposium. I also have to thank the Millennium Institute for Integrative Biology for also helping us out with having this room for the whole day, and the Institute for Biological Medical Engineering, who also chipped in organizing this symposium. And this is also framed in a project that it's a project in collaboration with Brazil, with the University of Sao Paulo. So it's a project between two people that are actually speaking today. So one of them is Beatriz Diaz, which is sitting over there. She's from the School of Biological Sciences, and she and I have been collaborating on finding plastic degrading enzymes in very silly extreme environments in which you might think that you cannot find them, but you actually can. And so we have this project for 2 years in which we actually studied with Brazil. We use structural biophysics, and we use enzyme characterizations and metagenomics to try to find new enzymes that can degrade plastics. So we had this project between our National Agency for Research and Development and the funders from the university, from the state of Sao Paulo in Brazil. And so we have to acknowledge all of that. The introduction for the whole symposium is going to be given by Mario Vera. He acts as a vice director of research in the Institute for Biological and Medical Engineering. So it makes sense that he's presenting what we're talking about today. And then afterwards, I'm going to be taking over to present the speakers. So I hope that you're excited as much as I am about the whole conference. I hope that you had fun. I also hope that you take either the opportunity after every talk to make questions, even though you might find them sometimes very naive. They might be quite interesting. And I will also suggest that you spend coffee breaks speaking with the speakers, because they are quite fun. And we had a blast this week. So they've been arriving during this week, and they're just fun and very good to talk to. You will learn a lot. So then I'll acknowledge people that came here for very, very long, which is Wolfgang Zimmermann from Germany, Başak Öztürk, sorry if I mispronounced that, also from Germany, and then Kara Lavender-Law from the US. And the organizers will be me, so if you have a doubt, just ask around. Mario Vera, and from the Institute as well, but also from engineering, and then Beatriz Diaz from the School of Biological Sciences. So thank you so much for coming, and then I'll just give the stand for Mario now.
Thank you. Good morning, everyone. I'm glad to be here to open this symposium, which has a very fancy name. Life in Plastic is not fantastic. This, as Cesar mentioned, this symposium would not have been possible without the support of the School of Engineering through the Institute of Biological and Medical Engineering. Today here we have also our Associate Director of Research and Innovation, Constance Flick, from the Engineering School. Also, there have been funds from the university through the Vice Dean of Research, Vice Director of Research, and also from our National Agency of Research, ANID, through the different programs like Iniciativa Científica Milenio, through the Millennium Institute for Integrative Biology, eBio, as well as a project that Cesar already mentioned that is also financed by ANID. Yeah. Also, this event comes with the support of two ministers, so the Ministry of Science, Technology, Knowledge and Innovation, as well as the Ministry of Environment. Yeah. Just to, although I'm not working in plastics, I'm a microbiologist and also work in microbial biofilms, so it seems too interesting to me to present a couple of slides to put this in context. Yeah? As you know, life these days is unable to exist without plastics. Plastics have become a major part of our environment. Yeah? And just on a timeline, as you see, the first plastics were discovered at bakelite, you know, this old plastic of telephones. Also, PVC and polyvinyl acetates were invented. Then in the 30s, polyethylene and nylon were patented. 40s and after the Second World War, there was a boom of production of plastics, yeah, and around the 1970, already were synthetic substances worldwide present, and as you see in this, this is also very funny that the men also, in theory, when they arrived to the moon, they put a plastic flag with a plastic material, so already escaped the planet. Yeah, and as you see, the concern of production, year production of plastics in millions of tons is rising and increasing, and this started to become a very serious environmental problem, and the last couple of years has been also some regulations of ban of one-use plastics through the European Commission, and also a lot of research has been ongoing in these last 2 decades about the fate of the plastics in the environment and the effects of the plastics on the ecosystems. Just to summarize, plastic debris comes from different sources. Basically, microplastics and small plastics are, you know, thrown by weathering and rainfall into oceans. Also, it's known that plastics that we consume and we throw about in our wastewater are also not degraded completely in our wastewater treatment plants. So therefore, we have also an income of plastics in our treated water. Yeah? So this debris can of course become small particles, and they can float on the environment, but the floating plastic or the surface plastic accounts just for around 1% of the global plastic amount. This means that this 99% they call missing plastic is in different forms, in different sizes, and also on the sediments. Yeah, so according to the density of the plastic and the size of the plastic, particles can settle, and the seafloor is full of polystyrene, polyethylene bottles. And the problem of being at the seafloor is also that they are less prone to degradation because there's no oxygen at the seafloor, very low levels of oxygen, and no light. So photochemical reactions are mostly happening on the photic zone. Yeah? And also, some plastics can be transported by zooplankton. So zooplankton can feed on plastics and move through the water column in order to change some fate of plastics on the environment. As you see, also plastics are providing a new surface for microbial life. So microbes can attach to different surfaces. As you know, microbes can attach to almost all surfaces. They form biofilms. And in some cases, also it has been detected and shown that several types of microbes can accumulate into the plastics. And the concept of plastisphere is already well accepted. So there's a new kind of surface provided by the whole amount of plastics in the environment, causing a lot of different changes on the ecosystems. Plastics can also be degraded by some microorganisms at very low rates. Yeah? So maybe microbial degradation will not be the solution for the whole amount of plastic we produce, but it's very good to understand the effects of microbial degradation on some plastics, because there could be leakage of some byproducts to the water column, and also some settling microbes can start to degrade the plastic, but also this plastic can act as a carrier for moving microbes through a larger surface. So plastic can act as hitchhikers for microbial life. Yeah. Last but not least, plastics are also providing surface for adsorption of organic compounds and hydrocarbons onto the surface. This means that plastics could bring also hydrocarbons together, and this plastic, of course, will go into the trophic chains by accumulating into microbes that go to zooplankton and of course to higher organisms, especially filtering mussels that can accumulate significant amount of plastics. So especially when we eat mussels, we are probably eating a certain fraction of microplastics into our bodies. Of course, fish can also eat plastics, but like we don't eat the stomach fish normally. So therefore, but it's already shown that some microplastics can pass the intestinal barrier and going into the blood and to the lymphatic systems. So the fate of plastics in animals is not well understood yet, and today we will have some more data from our invited speakers. So the problem is massive, and for sure the solution will not be accomplished in the next years, and so there should be an environmental policy change, a change on the production of plastics in the future, maybe through more biodegradable plastics, as well as a more complete understanding on what's happening on the microbial life and on the ecosystems because of the massive amount of plastics into the environment. So just to present our symposium, today in the morning we will have 3 talks, yeah? As Cesar will present them soon, so Cara, Cristobal, and Beatriz. And now I invite again Cesar in a couple of minutes to present Cara. So she will be our first speaker from the Sea Education Association in the United States. Then we'll have a coffee break. We are on time, so we expect to be on time for the coffee break. And we expect this coffee, as Cesar mentioned, to be an opportunity to exchange ideas and make questions. Then we'll have the talk of Cristobal about micro-sized plastics particle accumulation in different trophic levels in our country. I forgot to mention that the majority of studies of microbes and microbiomes and accumulation has been done in the northern hemisphere. And this is something we are missing. So in the southern hemisphere, we still miss a lot of research and data, and especially when the massive accumulation of plastics in the 5 big spots in the world are occurring, also one in the southern hemisphere, or 2, I think. Then Beatriz will talk about metagenomics for search of hydrolysis of polyesters in marine environments. So let's just to mention, some people have estimated that there are like 10 to the power of 21 bacteria in the oceans. So there's a huge amount of biodiversity and microbial enzymes remain to be discovered. Yeah, and metagenomics is a very powerful tool for this type of studies. Then we'll have a free time for lunch of 1.5 hours, and we go back here at 2 o'clock for our afternoon session. Yeah, where we have also 3 speakers. So our first speaker in the afternoon is Professor Dr. Wolfgang Zimmermann from the Institute for Analytical Chemistry in the Leipzig University, expert in enzymes and hydrocarbon metabolism for especially plastic degrading enzymes. Cesar will also give a talk about PET hydrolysis and some very interesting proteomic protein studies about them. And Basak in the afternoon will also talk about, sorry, I forgot the titles here, but after Wolfgang we'll have another break, the last break of the afternoon. And then Cesar is coming to talk about some engineering, protein engineering of PET Antarctic degrading enzymes. And then Basak, our last speaker today, will talk about biodegradation of aliphatic and aromatic products, plastic products on marine environments by microbial communities. So with this, we will make also a group photo and social mixer up to 6 o'clock in the afternoon. So now I will invite again Cesar to present our first speaker. And thank you for your attendance. And also there's an online streaming upon request.
All right, so we are almost all set for the first talk. Again, I'm pretty bad with introductions. I'm sorry about it. But I can mention that the next speaker who's opening this session is Cara Lavender Law. She's a research professor of oceanography at the Sea Education Association, and we actually had a really nice talk this week on Wednesday at the School of Biological Science about what they do, which is very different from what I'd imagined, actually. So the typical setup will be for undergraduate students, and this is a shout out for undergraduate students, to go sailing in the ocean for quite a few weeks, and then collecting samples in the ocean, and then doing some studies that they planned before traveling. And so I think that it's an amazing thing to do that hopefully we can do also in our country. Maybe we can travel there for doing it. So she's been working on plastic debris in the ocean for about 15 years. And that is because I will say personally, that's how long it takes for actually accounting for the amount of plastics that are present in the oceans. Which is not, it's not easy as it sounds. Like we have the reports now where we can understand that plastic is floating around, but then whatever we conceive that it's like in our imagination of what plastic debris is and how it looks like, it's quite different from the actual perception from a researcher in this area. She's been serving in many marine debris working groups in many different places, and she's well supported by different agencies, funding agencies in the US. And we met in Monaco during a conference about plastics last year, and she also opened the session, and it was an amazing talk. So it made sense for us to actually invite her to give this talk today. So she will be talking, like Mario showed before, and I'll just quickly pop into the titles of the presentation. She will be talking about framing the global plastics problem and environmental perspective. So thank you so much for coming, and you're most welcome to take the stage. Yeah.
And there's a picture on that too?
Yeah. Okay. So that's for plating.
Perfect.
And that's for switching.
Okay, thank you. Okay, well, good morning, everybody. Thank you so much, Cesar, for the kind introduction, Mario, for setting us off on a path that I will nicely follow with some time frame figures as well. I'm really excited to be here, and I'd also like to thank Cesar and all the funding agencies that have supported this symposium, because I think it's a very important conversation to have, and to have these conversations across disciplines. So I may be a little bit different in my expertise from many of you here, but I'm here to set the scene today by sharing my perspective through the ocean lens. So the global plastics problem, of course, is much more than just an ocean problem. But what I'd like to argue this morning is that without the ocean focus, I think we might not yet be taking seriously this very complex situation that we've created with plastics. We know that more than 70 years of innovation, as we just saw in that nice time plot, of this exceptional class of materials has brought really unprecedented societal advancement in many different ways. And with that, we've all become extremely reliant on plastics. Just look around the room. Look at what's in your bag and in your home. But we all know that this has come at a cost, and this is a cost that really I think we're only beginning to seriously address now. And the story I'm going to tell you today really begins with the cost at what I like to call the end of the pipe. And by that, I mean the middle of the open ocean. Oh. Okay. So I really have two main objectives in the talk today. The first is to talk about the science, from my perspective, what I think we actually know about the scale and the scope of the plastic pollution problem, with a focus on the oceans. And in that, I will present a fair bit of historical context as well. And then the second half of the talk, I'd like to present some food for thought, really, on potential approaches to addressing global plastic pollution with the goal of achieving what I call sustainable outcomes. And by sustainable here, I'm referring to collectively environmental, economic, and societal outcomes, recognizing that by optimizing one of these, whether the economy, the environment, or society, that we often have to accommodate costs in one of those other pillars. But to solve the problem, we really have to optimize all three. Okay, so you just saw this curve in a slightly different format on the left side. This is a curve of global plastic production, and this is shown as a function of time now since 1950. So this is the sort of nominal start of commercial production of plastics. And as Mario mentioned, there was almost from the beginning a very rapid growth in plastics production. I should say this is the blue curve. We're looking at millions of metric tons across time. And in fact, the growth in plastics production has now outpaced the growth of all other bulk materials, including steel and aluminum and cement. And global plastic production has more than doubled since the year 2000. That growth really reflects the nature of this class of materials that are now essential to modern society. And we all know the properties of synthetic polymers, including lightweight and durability, their strength, chemical resistance, combined with a breadth of chemical additives, makes these materials really almost limitedly versatile in their application. And their application can be both for short-term or single-use disposable type applications, or for long-term or more durable applications. Well, in work led by Roland Geyer, cited down there, we integrated this production curve over time, and we found that more than 8.3 billion metric tons of plastics have been produced since 1950. And that number is so enormous that I can't give it to you in terms of football fields or blue whales or Empire State Buildings. It's meaningless, the number of those items that we would need to accommodate for 8.3 billion metric tons. What we can see on this curve also on this plot is now a curve of plastic waste generation in red. And note, it's the exact same scale on the y-axis and the same time period. And you can see that the curve is very, very similar in shape and only slightly offset from the production curve. And that's because plastics pretty quickly become waste. Most of the plastics we use, we dispose of within a year of their use. The offset accounts for those durable items like our furniture and our laptops and our cars. Well, what we found in this study was that 70% of this 8.3 billion metric tons have already become waste, and only 9% of that waste was recycled, and only 12% has been incinerated. So what that means is the vast majority of plastic waste is residing either in landfills or somewhere in the natural environment. And when we take that number together with the plastics that are still in use, this means that 90% of the plastics that we've produced are still in existence on the planet today. Well, these are big numbers, and we can think of these numbers not only in terms of mass, but also in terms of carbon. So this is a figure from a paper published with lead author Aaron Stubbins that I was fortunate to be involved in. And he's a geochemist. And so his idea was to compare the amount of plastic on the planet in units of petagrams of carbon to natural stocks of carbon. So what we found was that by 1962 already, the amount of carbon in plastics had equaled the carbon in the living global human biomass. By 1994, that number equaled the global animal biomass. And what we see is that if production continues along the same curve, which is a projection, it's actually projected to accelerate, probably even faster than that curve. But if we follow this curve, that the carbon in plastics will equal the global bacterial biomass before the end of the century. And that's a big number. Okay. We often I want to show you this curve one more time in a different context here. So we often hear the term plastic as if it were a single material, whereas instead plastics are a material class that consists of hundreds or maybe even thousands of different types of distinct polymers that are further diversified by chemical additives. But when we break down the curve of global plastic production, so this green curve is just identical to the one I showed you before. If we break that down into polymer-specific contributions, we find that the majority of plastics, nearly 77%, of those produced to date have a common chemical structure, and that chemical structure consists of a carbon-carbon backbone. So the chemical structure is really important to defining the properties of the plastics, like those that I mentioned earlier, but it also makes these materials very strongly resistant to biodegradation. It's very difficult to break those carbon-carbon bonds. Well, if you're designing a material or a product, this might be a really desirable characteristic, especially if a product is designed to last for a long period of time. You don't want it to biodegrade. But the resistance to biodegradation also means that these materials can become very persistent when they end up in the natural environment. They're persistent, but they're not stable in their composition. We know that when plastics are exposed to sunlight and in the presence of oxygen, that photochemical degradation occurs, and this causes a reduction in the strength of the materials, and it makes them susceptible to breakage into smaller pieces, causing fragmentation. And we're going to come back to that point in just a little bit. Okay, well, the last point I want to make with this curve is that when we even think about these carbon-carbon backbone plastics, the packaging industry actually accounts for a large proportion of those plastics and therefore of total production. And we all know that these kinds of products, packaging and other single-use items, rapidly become waste. And so this is really what explains, at least in part, that red curve I showed you about plastic waste generation nearly matching production. Okay, we're now going to beat this dead horse and look at this one more way, this time scale. We're going to use the time scale now to look at the historical evolution of our knowledge of ocean plastics. So the first evidence of plastics contamination in the marine environment really was in 1966, when a paper was published. Very hard to see this image, but this is a picture of a seabird carcass. This is actually from the Northwest Hawaiian Islands. It's a Laysan albatross chick. And scientists in 1966 looked at 100 birds, and already 74 of those birds had plastics in their gut. And this was only when global plastics production was just 5% of what production levels are today. The first reports of microplastics floating, confused with this. Okay, microplastics floating in the open ocean was published in 1972. This was by scientists at the Woods Hole Oceanographic Institution. There were also reports in the early 1970s of plastics floating in the North Pacific, including large, really recognizable items like flip-flops. So this was when production was just 10% of current levels. So my organization, Sea Education Association, which I'll tell you a little bit about later, first started studying floating microplastics in the mid-1980s, and chief scientist Jude Wilbur published those first data in 1987. But I would argue that really it wasn't until the 2000s when people really started to pay attention. And that's when, in 2003, a sailor and an environmentalist named Charles Moore wrote an article in Natural History magazine describing his view of the North Pacific Ocean from the deck of his sailboat as, quote, "as far as the eye could see, the site of plastic." And it was in this article that the term garbage patch was born. So although plastics production had been on this really rapid growth trajectory for a long period of time, and scientists had been documenting plastics in marine life and plastics in the ocean for decades, really nobody was talking very much about plastic in the ocean until this garbage patch entered consciousness. So soon after its birth, the garbage patch turned into this sort of phenomenon of its own. It was being described as twice the size of Texas or 3 times the size of France, as having more plastic than plankton, or being a floating island visible from space. And these are very striking and very memorable sort of images, but I'm going to tell you, perhaps twice if you heard my seminar earlier this week, that there is no floating island of trash in the Pacific Ocean. Here's my first bit of data to illustrate that. This is the view of the eastern North Pacific Ocean in the region of the garbage patch, as taken from the International Space Station by a close friend of mine who was a graduate student with me, who is now a NASA astronaut and spent 6 months aboard the International Space Station. And she was very kind to take a picture of the garbage patch by request. You can see that really it is just water, and in fact, most of the time, clouds. You have to trust me, given that there's no land masses here, that this is actually that part of the ocean. Okay, but we do have some more firsthand evidence here about what this part of the ocean looks like from the deck of a ship. So if you're sailing in the subtropical latitudes of the North Pacific Ocean or the North Atlantic Ocean, where I've personally sailed, but I suspect also in the South Pacific, in these so-called garbage patches, this is likely what you see most of the time on a nice calm day from the deck of the ship. Right? You see water and clouds, maybe you see some seabirds or some marine mammals. Really rare to actually see a ship. But you will see things that you don't normally see floating, especially if you are someone who has done a lot of open ocean sailing as a sailor or as an oceanographer. And you do see floating debris items drifting by, perhaps one item per minute, quite frequent. And they might be recognizable, like this boot that's floating upside down or this 5-gallon bucket. Sometimes you can see really disturbingly familiar items like food packaging and this egg crate or this tire. This is rare because these items were observed a year and a half after the Japanese earthquake and tsunami, which was of course a very tragic human event, but also put millions of tons of debris of all kinds into the Pacific Ocean. And a year and a half later, we could see very clear evidence of items that had entered the ocean from that particular event. But most of the time, the items you see are kind of unidentifiable. You don't know their history. There's sort of large chunks of bits of plastic or line or film or masses of fishing gear that are drifting around. And when you see these items, regardless of what they are, it's really kind of exciting. Oh my gosh, what is that tea kettle doing or that toothbrush? And then it's shocking, and then really you're kind of depressed because who wants to see trash floating thousands of kilometers from land in the middle of the ocean? But the picture changes quite a bit if you measure what's floating at the sea surface by towing a plankton net through the water for just 30 minutes and sifting through its contents. And this is what thousands of our undergraduate students have done for nearly 40 years in our sea semester program. So as Cesar mentioned, we are a program in the US. We mainly draw from United States colleges and universities, but we're increasingly looking for international students. Our students come and study with us in Massachusetts on our campus for a few weeks where they prepare to sail on oceanographic research vessels that look like this. We have one that sails in the Pacific and one in the Atlantic and the Caribbean. And they design research projects that they will carry out on board the ship where they are not only oceanographic research scientists, but also crew members, so helping to operate the ship. Happy to talk more about that at the coffee break. So our students have been deploying nets like this. It's about 1 meter wide, 1/2 meter tall at the sea surface, right at the air-sea interface, twice a day for nearly 40 years. And what you will see when you pour the contents of that net into a sieve, among the plankton and among the organic matter, which are sort of the clear and brown blobby bits, are bits of what we now call microplastics. And most of these microplastics, I should say the mesh of the net is 1/3 of a millimeter in size. So the holes are small enough. They're quite small, but you can still see them. So most of the particles are large for what we think of as microplastics, perhaps, but they're very easily definable because they're brightly colored, they're irregularly shaped, and if you pick them up with tweezers, they're quite hard. So it turns out that based on a laboratory analysis back at our labs of more than 4,000 particles, that we can say confidently that the human eye can pick out plastics from natural organic matter at these sizes. So sizes larger than 1/3 or 1/2 of a millimeter. Well, in the 15 or so years since I've been working on ocean plastics, it's become readily apparent that microplastics are the most numerically abundant form of plastics floating around in the open ocean. And in fact, the most widespread type of debris found in the marine environment more generally. So you can see there are images here of microplastics mixed among volcanic beach sands in Hawaii. These were floating in the water column. We've even found microplastics buried deep in ocean sediments, as well as frozen into Arctic sea ice. And in this case, you can see some of these microplastics that are actually more fiber-like, that are likely from textiles or possibly from fishing gear. So we've learned that microplastics are abundant and widespread in the marine environment, but they are also abundant in the freshwater environment, in lakes and rivers and streams, and in the terrestrial environment, in soils that are agricultural or otherwise. Microplastics have been found in the air, in snow, in precipitation, in drinking water, in bottled water, in groundwater, in wastewater, in stormwater. It seems that the smaller the particle, the more abundant and widespread it is. And that is especially true for things like these microfibers, because microfibers can not only be readily transported in the water, but also in the air. So this is some work from Janice Barani at Utah State University from 2021, where she looked at wet and dry deposition in US national parks in the West. And she found that between 2 and 6% of particulate aerosols were actually composed of plastics. She then led a follow-up modeling study to assess the likely sources of these atmospheric microplastics. that were deposited to the Western US. And you see that some of those illustrated here, which I'm not going to go into in detail. But she also led a modeling effort to try to predict patterns of where you would expect high atmospheric deposition of microplastics. And I show this just to indicate that that pattern is not uniform everywhere. It is not uniformly sort of settling microplastics across the globe. So this is also true when we look at the ocean. So here's a map of data collected by our students since the mid-1980s in the Western North Atlantic Ocean. So here we see North America, South America, here's the Caribbean Sea, here's Bermuda. And what you're seeing in this map is each dot indicates a single plankton net tow. So I put a picture here to remind you how we're collecting these data. And the dots are colored according to the measured plastics concentration in units of pieces per square kilometer. So the students count the plastic and divide that number by the surface area that was sampled by the net. So the darkest blues are 0 plastics, the warm colors are the highest plastics. And to give you a sense of scale, the highest values here are in the hundreds of thousands of particles per kilometer squared. This Petri dish contains about 240 pieces of plastic from a single net tow. And when you scale that by the area sampled, that is equivalent to about 130,000 pieces per square kilometer. So a big number, but not a big mass of plastic. You could easily hold that in your hand. Okay, so if we look at the distribution of plastics here, what you can see is that the highest concentrations, which are these stars, are found far offshore, which is a little bit counterintuitive. And also the highest concentrations are pretty clearly located in this band of latitude between 20 degrees north and 40 degrees north. And this can be quite easily explained by ocean physics, even theoretical ocean physics that's then been demonstrated with data and with models. So there's the ocean gyre that carries material around the ocean basin, but the currents kind of come together and slow down right around 30 degrees north. And this is what I call the dead end of the ocean, but this is really what the garbage patch is, right? This is this high accumulation of floating microplastics. You can see if you look that not every tow in this high accumulation zone or in the garbage patch has plastic. There are actually plenty of zeros, which are a little bit buried by the way the map was constructed. And you do see high values outside of that zone. But by and large, there's this very clear pattern that can be well explained by ocean physics. So we can actually just drive that point home again by looking at our data collected again from our students, but now in the Eastern Pacific Ocean. So here's North America, Hawaii, and Tahiti, Puerto Vallarta, Mexico. I like to show this because you see a much broader latitude range and you see the same accumulation in the subtropics for the same ocean physics reason. But you can see now that really outside of that region, you see very little plastic floating at the surface. And that's because the ocean physics are pulling material away from the equator, away from the tropics in what we call a divergence zone. So even though we do see sort of high variation really throughout, you can see tows with a lot of plastic next to tows with almost no plastic. By and large, these patterns are well explained by ocean physics. Now, there's an important point to know when we're going out and sampling the environment, and that is that the data that you collect are highly dependent on the way that you sample. So in this next slide, this is here to remind you the size of plastics we're measuring in the net. This is in contrast to the next slide, which is a project led by a citizen scientist effort through adventure scientists. And in this case, people were asked to take a 1 liter water sample from wherever they are in the world, could be freshwater or seawater. And then the technicians actually filtered that using either a vacuum pump or a hand pump onto a 0.45 micron filter. So the smallest particle that could theoretically be measured using this method is actually 750 times smaller than the smallest particles that we would collect in our plankton nets. And if you look at the distribution, so here actually I'm showing you on the upper top, most of the particles that they collected in this effort that you see was a global effort were actually those very fine microfibers, which we do not collect or we don't quantify in our nets. So if you look at the patterns here, you can see the bubbles are scaled according to the amount of plastics that they found in plastics per liter. The numbers ranged from about 1 or 2 particles per liter to more than 500. And there's also plenty of samples that had no microplastics. So when you look at this, you really don't see the same pattern that I showed you from the plankton net data. And that's because these are much finer particles that are not just being carried by ocean currents. This is where the atmospheric deposition also likely plays a role. So when we think about the two slides together, there are a couple things to remember. First, a microplastic is not a microplastic is not a microplastic. At the very base level, there's a huge variation in size, and these may be even as small as nanometers in scale. But they're really not globally distributed evenly and not in the same amounts everywhere. Okay, so why do we talk so much about microplastics? Well, certainly from an environmental science perspective, we're concerned about the impacts on marine life. And especially for microplastics, I think the primary concern really is around ingestion by marine organisms. So if you think about large animals like whales or even the seabird I showed you at the beginning, they can ingest quite large items. I mean, things like flower pots and car parts have been found inside whale carcasses. Plastic bags have been found in sea turtles, and cigarette lighters and bottle caps in seabirds. But when we think about microplastics, these are small enough to be ingested by much smaller organisms. Not only fish, like you see here with sort of plastics of the same size we collect in our plankton nets, but also barnacles. This barnacle was living on this plastic buoy here, but even down near the base of the food web where zooplankton have been found to have ingested microfibers and very small microplastics. So these are really accessible almost to the entire food web. To date, plastics ingestion has been reported for more than 1,200 marine species, and even more if you go beyond the marine environment. And we know that at least for larger microplastics, the sort of physical contamination of the animal can reduce the stomach volume of the animals. It might even cause internal damage if they're sort of larger or sharp pieces, and animals can feel falsely satiated and stop feeding. But when we can, some of the other impacts of microplastics that have been shown are things like inflammation or other sublethal effects. And as Mario mentioned in the beginning, even potentially the transfer across cell membranes, or across, tissues. So we do know that in some species, the frequency of occurrence of microplastics ingestion is very high. And a prominent example is that 95% of northern fulmars, seabirds in the North Sea, that have been examined have had plastics in the gut. But even given that high incidence, we're not sure what the impact is on the population. We know that the plastics ingestion alone, the particles, can pose risks to marine animals, but this is complicated even more. Potentially the risk is even elevated because of the chemicals that are associated with these plastics. And these can be chemicals that are byproducts of manufacture or that are deliberately added during manufacture, like those chemical additives, or they could also be sorbed to the plastics if they are already present in the environment or in seawater. And there is evidence in many of these cases that the chemicals themselves can cause harm to organisms. The concern, of course, is that if the animal ingests the plastic particle, that these chemicals might transfer into the animal tissue after they've been ingested. And there have been quite a few laboratory experiments that have shown for very specific animal and specific polymer and specific chemical combinations that harm can occur. Well, given the complexities of the microplastics themselves and the diversity of marine life and the variation in exposure in the environment to microplastics, what does the weight of the scientific evidence indicate about biological impacts of plastics or microplastics? So what I'm showing here is a fairly complicated set of figures from a 2019 critical review led by Chelsea Rockman's group at University of Toronto. And the goal is to evaluate the evidence published in the scientific literature on the impacts of plastic debris ranging in size from nanometers all the way up to kilometers. So on both of these plots, you see the x-axis indicates the size of the debris that was evaluated. And then there's the level of biological organization from ecosystem level to organism level, all the way down to suborganismal level, even into small molecules, atoms, etc. So what they did is they investigated the literature, and each box here indicates a level of biological organization for a particular size of debris. And the study then tested particular effects, and then they reported them as either detected or as not detected. So the darker the square, the more effects were tested and detected or not detected. Okay, so if we look at the part of the figure that's specific to microplastics, so we're looking at millimeters and smaller, what you can see is that there were 400, if you add up the numbers in the boxes, 421 effects were tested. And of those 421 effects across all of these levels of organization, a little bit less than half were detected, and a little bit more than half were tested but not detected. So this suggests, of course, that there's reason for concern. There is evidence of biological impacts due to microplastics at a variety of biological levels of organization. But you can't yet make sweeping generalizations about microplastics are killing the ocean or microplastics are killing humans, right? And this is perhaps never going to happen because there are many inherent challenges in this kind of work. First of all, there's variation probably among studies, but you know, by and large, these studies were also in controlled laboratory conditions where often the animals were dosed way higher concentrations than you would ever find in nature. That is starting to be corrected now in the literature. But in the environment, if you try to take your lab results and try to figure out what's happening in the environment, that's really hard because the animals are likely exposed to complex mixtures of particles and materials, and their concentrations are highly variable, as I showed you in some of the earlier maps. And so, and even in many places, we don't actually know what a particular animal or a population is even exposed to. So there's a long road left to go to sort of figure out maybe what are the most harmful particles or chemicals or polymers and to whom. But this is where a huge amount of the work is actually going. I just want to turn your attention to the large side, the macroplastic side of the plot now. So this is for plastic debris larger than millimeters up to, say, kilometers. So think like large fishing nets. In this case, actually, the weight of evidence is pretty clear. It is, first of all, most of the studies are looking at the tissue level and above, up to ecosystem level, and most of these studies have found clear effects of plastics. And this is really not surprising because we've all seen these images that really drive emotional responses. So this is the modern day poster child of plastic pollution in the marine environment. This is an albatross chick, again, from Midway Atoll in the Pacific, and here's that cigarette lighter and those bottle caps. But there are many of these kinds of images that we've probably all seen. And I would argue that it's not only that we had sort of this image of floating islands of plastic that got the public really upset, but I think also it's images like these that, of course, have created sort of visceral reactions to the problem. So I think in part, these are sort of the two parts of why we're all here today talking about ways to try to solve this global plastics problem. Okay, of course, the next piece that's going to keep this in our attention is that now we're talking an awful lot about human health. So there are now a wealth of studies that are documenting microplastic in items that we consume, not only seafood, but bottled water and tap water and sea salt and beer, honey, tea, the list goes on, even fruits and vegetables. So we know that we're consuming these microplastics in our own diets, but are they actually in our bodies? And now we're seeing studies coming out pretty rapidly that indicate, yes, there is exposure and transmission of microplastics in the human body. And the question really remains, much like in the marine animals, are these causing harm? So we can't answer that question yet. Most of these studies are indicating contamination of humans, but not actually answering the question, what happens when we have these in our bodies? But I think, again, for this reason, attention to the plastics problem is not going away, and it's likely to intensify. Okay, so I want to take stock of the path we're on before going on to talk more about what we're going to do about this problem. So what do we actually know? We know that billions of metric tons of plastics exist on the planet, and there's pretty clear evidence this number is likely to grow and even accelerate. We know that most plastics, those with carbon-carbon backbones especially, are strongly resistant to biodegradation, and therefore they persist in the environment. They're going to be here for quite a long time. We know that plastic debris is widespread but not uniform across the globe, and it spans a tremendous size range from microns and possibly nanometers to more than hundreds of meters in size. And plastic debris harms wildlife. Okay, now there's the what do we sort of know. So I haven't really had time to talk in detail about plastic inputs into the environment, including the ocean. But this has been estimated by multiple studies now to be on the order of tens of millions of metric tons entering ecosystems annually. The challenge is that most of these are modeling studies and really based on little to no field data. So we have very few instances where we have directly measured the flux of plastic debris to air or water or soil. And where we have, those measurements have been extremely variable. So I would say approach that number with caution. It's really intended to be an order of magnitude estimate. We sort of know that plastics are readily transported by air and water, and they're transformed when they're exposed to sunlight, heat, and oxygen. But those rates of weathering and fragmentation and the mechanisms causing them in the environment are quite poorly constrained. And that means we don't really understand the resulting particle size distribution or the time scales of persistence and the ultimate fate of these plastics. And then contamination of biota by microplastics, and I'd say by humans as well, is widespread. But risk assessments are underway, but they're really still in their infancy. So I think it's really difficult to quantify or even qualitatively determine what the scale and scope of the impacts are due to microplastics in the natural environment. Okay, so there's still a lot of work to be done on the environmental side. But I would say, and I think almost all environmental scientists will agree with me, that we now know more than enough from an environmental perspective to take action. So this brings us to the next part of the talk, which is trying to think about the problem and how we're going to address it. So I've titled the talk Framing the Global Plastics Problem, but as you of course know, I'm framing this through an environmental lens. And so the problem as I'm posing it here is that plastics pose a threat to environmental health. And there are other reasons that this pervasive global plastics problem is a problem, and there are impacts of things like plastics manufacture and use and disposal on, say, carbon emissions or energy use or economics. There are environmental justice issues around all of these things. And those are absolutely important to consider. But here we're going to restrict this to thinking about the environment. And so therefore, I'm going to pose the solution as, well, we just need to keep, forget about those for the moment. We need to keep the plastics out of the environment. That would be one of the most direct ways to solve the problem as it's posed here. So what I'm showing here is one of many frameworks that's been proposed to prevent plastics from leaking to the environment. And this one was put forth by a committee that I served on at the National Academy of Sciences in the US that presents interventions beginning at the production phase of plastics. And this goes all the way down through design and use and disposal or loss to the environment. So many actions have been proposed in sort of each of these steps along this chain, and many have even been taken. And I'm going to take a minute just to describe a few of these to give you a sense of what we're talking about here. So first, here back to this curve that we've seen 100 times now. I'll tell you that there is some evidence that this curve will accelerate because the fossil fuel industry will need to start pivoting away from providing fuels as we electrify our grid and our transportation into using petrochemicals for other things, including plastics. So the concern is that this is really going to start accelerating even faster. And there are many, many calls that this curve needs to be capped or even reduced. We can talk in detail about how likely that is, how you would even go about doing that. But this is one way that you can address the problem. Let's just not make so much plastic. Okay, the next category is innovate designs and materials, and I would say products as well. So this is an example of something that Coca-Cola just announced fairly recently, a couple of months ago, that they would be changing their canonical Sprite bottle from green PET plastic to clear PET plastic, and that is simply to improve the recyclability of the bottle. It's a lot easier to do something with clear PET than from green PET. So this is an example of a private company taking the initiative to actually attack their own marketing campaign, right, and make a change for the good. Next, decrease waste generation. So lots of ways we can decrease waste generation. There's a bill in New York City that now prohibits food service establishments from automatically including plastic utensils. You can also think about things like straw bans or bag bans, ways to sort of force people to use less and make less waste. And of course, we can all take the initiative to do that ourselves by using reusable items or just simply choosing to buy less, not buying a new iPhone every year or a new laptop every year. The less we buy and consume, the less waste we'll ultimately make. And then we absolutely need to improve waste management to keep plastics out of the environment. So, you know, we can, obviously this is a very local and regional issue, the way that countries and municipalities manage their waste. There are pretty extreme cases. This is a photo of a river in Vietnam where the river is the waste disposal method. You put your trash in the river and it gets carried away. But even in high-income countries where we have robust infrastructure, we have issues and we have leakage of these materials to the environment. So when we talk about improving waste management, this really spans all countries around the world. Just the way that you improve that is going to be very dependent on where you are. Capture waste. So this is sort of the last chance effort to prevent it from getting into the environment. This is a picture of Mr. Trash Wheel, who is a solar-powered trash capture device in Baltimore Harbor in the US. And you can see there are these floating booms that are capturing all the debris that comes down the St. John River. It's getting funneled into this conveyor belt and getting put into a dumpster so that it's being captured before it goes out into the ocean. And they're actually doing a lot of characterization of what's in that debris field to try to understand how much waste there is. And then lastly, you know, trying to minimize at-sea losses, and fishing gear is really the poster child for this. We know this is a huge impact to animals in the marine environment. It's really hard to quantify how much fishing gear is being lost, but this is sort of preventing those losses in the ocean itself. Okay, so that's all well and good, but how do we actually begin to prioritize such actions? We can't do everything everywhere all the time. So I have some thoughts, and then I'll wrap up about how we might prioritize these actions. So first, we might say, well, really, we need to figure out what's doing the most harm in the environment and start there. So let me say, these images are purely illustrative. This is not based on any scientific assessment of what's harmful, but just to give you some ideas of how we might think about this. Starting at the high risk, I mean, I think we can agree that this is a high risk situation for this sea turtle or the six-pack ring around this duck. So maybe we try to, you know, attack these types of items or uses of plastics. There's also, of course, concern that the smallest microplastics or microfibers might be the highest risk to organisms, including humans. And then as you go down, you know, here's this fish that had this plastic in its stomach, but as Mario said, we don't usually eat the stomach of the fish. And in fact, many of these plastics, if left alone, might have just passed right through the fish. And this is a very cute and disturbing image, but I'm not sure the seahorse is actually being harmed. So this is actually even harder to do when you start adding humans to the calculus of level of harm. But that really brings me to my next point of, okay, even if we agree, let's just tackle the harmful stuff first. Well, are we talking about the harm caused by a specific item or product, like that six-pack ring? Is it a specific polymer or a specific plastic, maybe even the additives in the plastic that are causing the most harm? Is it the particle characteristics that are causing the most harm? And then harm to whom? If we find clear evidence of harm to humans, I think that will motivate the fastest action, but it could be harm to ecosystems that provide human services, or to specific species that are commercially important or threatened or endangered, or that are culturally important. So, you know, even if you agree that we need to do this by thinking about harm, I would hope that people would involve, you know, environmental scientists and toxicologists to collaborate to sort of understand how to quantify that through tools like risk assessments, for example. But I think in the end, many of these questions, especially on this side, are going to end up being value judgments. Okay, we could also try to prioritize actions according to their feasibility of reducing or eliminating them. And this is what I call the low-hanging fruit approach. So just deal with the easy stuff first. And at least in the US, we were able to ban cosmetic microbeads and wash-off cosmetics, I think now 6 or 8 years ago. And interestingly, the manufacturers did not fight that. They said, okay, we'll use ground up nutshells or something instead as our exfoliant in our microplastics. So, the easy to eliminate are sort of unnecessary products, bag bans and straws. That's why you see these things, I think, having regulations around them already. But it gets harder as you go up the curve, right? So let's think about the water bottle. There are municipalities in the US that have banned the sales of single serve bottled water because they want to eliminate the plastics. But not everywhere can do that, right? Not every place has potable water coming out of a tap. Sometimes there are emergencies where you need to bring in large amounts of bottled water. So this can be very difficult. Similarly with small sachets that many people purchase because they can't buy their products in larger volumes simply because they don't have the resources to do so. So there are innovators who have started to tackle some of these problems. And a good example is Algramo here in Chile, who's working on that. But then when we start going up here, tire wear particles are thought to be a huge input of microplastics to the environment and also quite dangerous in terms of their impact, or textile fibers. And I don't know how you eliminate tires or textile fibers. So this is, these are challenges. Another point though is that, who gets to decide something is unnecessary or who or where. So again, value judgments are laid in here. Okay, and then lastly, another approach would be according to time scale of implementation. So, there's some things we can do today. If you have a washing machine at home, you can install a filter that captures the water coming from the washing machine that will filter out those textile fibers that are coming off your clothing so they don't enter the wastewater that goes into the plant and that might escape into the environment. So that's a fairly cheap and easy way to at least deal with part of the problem. Sometimes I think, improving the waste management, it might be very expensive, but at least, putting a garbage truck in Vietnam is a straightforward way to at least start to deal with the problem. As we start going into things that have longer timescales, though, some of these are more policy oriented. So in the US and a number of states listed here, we have these container deposit laws where you buy your beverage and you pay an extra 5 or 10 or 25 cents, and then you get that money back when you return the bottle. And that really incentivizes the collection of these materials for recycling. There could also be post-consumer recycled content requirements saying you have to make your new bottle out of 25% recycled material that could perhaps sort of enhance the recycling infrastructure. And not that the recycling infrastructure is perfect, of course, and that's where we get into the sort of longer time scale of implementation activities. And I think those may be some of the activities most relevant to this audience. So in this category, I'm thinking about enhancing the processing of the existing plastics that we make, those carbon-carbon polymers and products, and that could be things like engineering innovations to improve sorting for mechanical recycling, but it could also be product redesign by making less complex products that have fewer materials that are very difficult to separate. But it could mean developing methods beyond mechanical recycling to break down existing polymers. It could mean designing new materials, not just for specific applications, but with end-of-life management specified and built into that design process. And really the end of this pipe, I think, is striving for circularity with minimal persistence of materials and harm if they are lost to the environment. So these activities take a lot of time, not only because of the long and winding path of research and development, but also because ultimately they're going to require robust data-driven analyses to evaluate their likely success in terms of environmental, economic, and social criteria. And I think in many ways, the timescale of implementation might be inversely related to the scale of effectiveness. I mean, if we can reach the holy grail of circularity, that's obviously going to be far more effective than simply putting more garbage trucks someplace to collect the material. So similar to the other proposed approaches, I think you can ask the question, who gets to prioritize? How should resources be allocated for research and development into new materials, or into new breakdown or recycling or upcycling processes? I would argue that we should think about whether the outcome, the new chemistry, the new enzyme, the new biology addresses a priority need. So is there a particular material that we would do well to substitute with a new material? Is there some way that end-of-life management will be much better for those high priority materials or products? course, you have to define what those high priority materials or products are. Other questions, can it be scaled, both technically, but also economically? And then importantly, of course, from the environmental perspective, we need to avoid regrettable substitutions. So I think we need to do evaluations of materials and products and processes to gauge whether there are actually improved environmental outcomes. And that might be measured in terms of carbon emissions or energy use or environmental impact. So there are tools like life cycle analyses, but I think we really need to be developing even more complex ways to analyze these. I think it is imperative that scientists and engineers that are working in this space consider this context. So I'm really pleased to be speaking to you here as an oceanographer, sort of setting the scene for the day. I think it's important to consider the context that, of course, I think my context is important that I've presented, but also to think about the specific aspect of the global plastics problem that your work is trying to solve. And I think it's equally important to be able to communicate how this can be a viable solution. And that's because policymakers right now, today, yesterday, are looking for solutions. And they're going to be making decisions whether or not they have scientific engagement or scientific evidence to back those decisions. So you may be aware that a year ago, UN member states passed a resolution saying we need a global agreement to deal with plastics pollution. And in fact, now there is a negotiating process happening. The first meeting of the INC was late last year in Uruguay, and the next meeting is coming up in Paris. And I was fortunate to attend sort of as an observer remotely for the first session. And I will tell you that there were calls from member states, from other delegations, NGOs, that the policy needs to be based on sound science. The challenge is it's very hard to even figure out what the questions are that need to be answered to make a policy of this scale, given the complexity of what I've presented today. So I think it's really important to engage all the different sectors who are thinking about the plastics problem. And in conclusion, I hope that you are inspired to join in, because there really is so much good work to be done and to be shared and to be applied so that we can create sustainable long-term solutions to the global plastics problem. Thank you.
Okay, so there's time for questions. It can be in Spanish and I'll translate, or it can be in English, whatever you feel like doing. If you have a question, just raise your hand, and there's two people on the background that are gonna give you a microphone for that. But I'll start with a quick one, which is about what you mentioned about timescales and also public policy. And I wanted to know your personal opinion on whether, because you mentioned that we need time for doing sound science, for finding out like what are the psychological effects of plastics, but then the decisions are taking place now. So are we running out of time for doing that science? Is there still time for like hopefully then deciding or adding different things into public policy so that we can change that trend afterwards?
That's a good question. The negotiation process is actually on a very short time frame. They are supposed to come up with an agreement by, I think, 2024. So it's that tells you sort of the feeling of urgency about the problem. But I do wonder if the policymakers recognize that, you know, going back to our timescale plot, it's really, although this plastics problem has been around for decades, I think the science has really only gotten started in the past 10 or 15 years. It's really accelerated. I feel like we hear about plastics all the time now, but even 15 years ago when I started, it was just garbage patch. Nobody was talking about, well, I don't know, I should ask if people were talking about enzymes that could break down plastics, you know, in the natural environment. So I'm not sure the answer to that. It's never too late. Absolutely, it's never too late. And whether it needs to be an adjustment by people trying to make the policies to sort of wait for some of the more long-term solutions, wait until the science can unfold. There's plenty to do in the short term, right? I think there are plenty of things that people could agree on today. For instance, collecting robust data on plastic production and waste generation and waste treatment in countries around the world. That very basic information is really not reported in a consistent, easily tracked manner. And hopefully that's something that policymakers could agree on while figuring out whether, PET is the material we should continue making, or if we should shift to other different types of more bio-based or biodegradable materials. So I don't know, but I hope that they will take the science as it is available.
And I also wanted to add on to that. I wanted to ask, how difficult was it to quantify the amount of plastics that's been produced and that ended up as waste? Because you just brought up that we need to enforce the way that we're quantifying plastic waste and plastic production, but for that article and for that research, like how long did it take and how sketchy the data was for actually finding that out?
Yeah, so I mean, that's a really good question. So Plastics Europe reports numbers on global plastics production every year. So that was an easy source of data. But it's, we were looking for also other types of data. So for instance, textile production. And a lot of the data came from sources where you had to pay large amounts of money to get access to the data. So in some cases, the data exists, but it's all proprietary, and it's not these industries are not willing to share it. And of course, there's gross assumptions that are being made. That study was still a modeling study. It was not a sort of rigorous, standardized data collection effort. And then as you go down the line to sort of the waste side of things, it gets even sketchier. And a lot of that came from compilations of data from the World Bank, for example, where countries self-report. So you can put a number on anything, right? You can make a number. How it's the level of uncertainty and the quality of the information going into that model that are really important to know.
I have two questions. The first one is about there are quite a few companies now around, or initiatives, which are focusing on removing plastic waste from the ocean via different means. I think there was one in Netherlands, for example, gained a lot of publicity. I'm always a little skeptical when I see these because I think they get a lot of publicity and I think it's good to raise awareness, but I'm not entirely sure about the efficiency or feasibility of doing this. What is your opinion on this?
Yeah, thank you for that question. I think, I put that, the colorful arrows of the interventions, and there was capture waste from the environment was towards the end. So I think trying to collect plastic and waste from the environment is always going to have to be part of our solution set. That being said, you may remember I said I'm giving you this talk from the perspective of the end of the pipe, which is the middle of the open ocean. And in the middle of the open ocean, most of what you're finding are pieces of plastic smaller than your pinky fingernail. If you think about that Petri dish where I said you could hold all that plastic in the palm of your hand, that was from 1 mile of sea surface that was being sampled. And in fact, if you translate that to just a particle per square area, it's like 1 piece of microplastic per square meter. So from a feasibility or efficiency standpoint, no, I don't think that is where we should be going to collect plastic from the environment. So there are some exceptions. There are also groups going out and trying to find enormous masses of derelict fishing gear, which of course are really harmful because they can continue ghost fishing and have very severe impacts on marine life. That's still very resource intensive to take a ship out to the middle of the ocean and try to bring back these many tens of tons of fishing gear. But I think from my perspective, a cleanup effort like Mr. Trash Wheel is a much more sensible way to go, especially because most of the plastic that is ending up in the ocean is most likely coming from land. And targeting larger items rather than microplastics, I think, is important. A caveat to that might be if you are a wastewater treatment plant manager and you want to figure out how to get more microplastics out of your effluent, right? So it is a balance. I think you have to sort of, but I think that's exactly the point, is what are the resources required and how much is it actually going to accomplish?
My second question is about, first of all, the definition and also the characterization of microplastics, because I have the feeling when I read publications that everybody has their own methods for, especially characterization seems to be, I know it is very difficult, but is there any effort to standardize what is microplastic? How are we going to classify the different kinds of microplastic so that, when we generate data, that at least this kind of metadata is uniform and they have been collected in a similar manner, for example?
Yes. The answer is yes. So the advantage of the historical perspective is I can tell you that most of what we knew about plastic in the ocean came from opportunistic data. I was towing a net to collect plankton and oh my gosh, there's plastic in there, right? And that's not, if you were to start from scratch, that is not how you would design a method to sample for microplastics in the ocean. So even though most of the literature reports things like this and everybody has their own net and their own way of counting and characterizing, yes, there's a tremendous push within the environmental community to try to standardize methods to the extent possible, but especially reporting. At least please, tell us how you collected the material. Was it a 1 liter sample? Was it a plankton net? And especially when we think of the microplastics smaller than those ones in our plankton net, you have to use chemical characterization to identify them as actually synthetic polymers, because especially for fibers, for example, there may be cotton or wool or rayon or other types of materials. So there is a huge push. We're not there yet. Of course, I don't know how you get a global scientific community to agree on a certain set of standards, but there are instances where we have done it before, and certainly in oceanography. So yes, that is highlighted as a priority.
We have a question on the back. If you can introduce yourself and then go with the question.
Yes, hello, I'm Gabriel Garrido. I'm more on the side of molecular biology, but I'm still interested in this. I have two questions too. It's the first is more or less on this line. How do you characterize exactly which plastics are in the environment, and how, for example, why haven't you used mass
Spectroscopy.
Yes, thank you. Difficult word. Yeah. To identify the chemical composition of this compound.
Yeah, so it's a great follow-on question to the last one. So the methods that are commonly used are spectroscopy methods, so Raman spectroscopy and Fourier transform infrared spectroscopy to identify the polymer type. And of course, you have to have robust libraries of polymers and additives and such to identify the chemical composition. People are using mass spec. There is a sort of push-pull in the community about whether it's more important to identify particles or more important to identify bulk chemical composition. So when you do mass spec, you lose the particle information. And if you do the Raman, it is extraordinarily time and resource intensive. Literally, most people are first visually identifying particles that they want to characterize. So there's already a bias there, and that there's an eyeball and a brain deciding which ones to test. And then you're doing it individually. There is a group in Germany that has developed an automated scanning technique, but it's quite expensive and hasn't really been adopted broadly at this point. So I think we're definitely seeing more and more work on that side of the characterization. Yeah, and you'll see mass spec as well.
And also, have you seen? You mentioned air pollution carrying microplastics. Have you seen like a difference in composition between the ocean microplastics and the air microplastics?
Oh, that's a good question. I don't know that we have enough data on the atmospheric deposition. So from our big pieces we're collecting in the plankton nets, we by and large are seeing polyethylene and polypropylene, and that is common across that size range. And we know that these are materials, the particles are probably broken down from our everyday consumer goods that I showed in that packaging slide in the beginning. When you get down to the really small plastics, you see a much greater diversity of materials. And I would say that there probably are not enough data to say whether there are patterns in what's being carried in the atmosphere versus carried by water, with the exception of microfibers, which you can probably at least narrow down what's being produced as a textile or as like nets and things like that and fishing gear, which is where I think most of these fibers are coming from. But I don't think we have enough data to really do that comparison yet.
Okay.
We will go with the last question.
Can I have a question about, so the 5 biggest contaminant countries for plastics in the world are all in Asia: China, Indonesia, Philippines, and so on. So how all these recycling politics can be established there? Because I see when you show the data, I can imagine how big are the numbers in China's oceans, but probably there's no reliable data on it. So
yeah, this is a hard question to answer. Yes.
The US produces a lot of plastics,
yeah,
but it's, as well as the European Union, are not the most contaminated places still.
Well, okay, so I hope we have a couple minutes for this one. Okay. So in the things we sort of know, I said we estimate tens of millions of metric tons of plastics are entering ecosystems. That work started with a paper led by Jenna Jambeck, of which I'm a co-author. And that's the paper you're referring to of the 5 countries contributing the most plastic to the ocean. So this paper was published in 2015. And it was an example of a paper where we really had no like hard data. This was all coming from the World Bank, who had compiled self-reported reports, right? And it was a report on waste globally. It was not a report on environmental contamination by plastics. So we took the data from that report and turned the crank, and what we found was the places in South and Southeast Asia that rose to the top in terms of potentially contributing the most waste to the environment were that way because the waste management infrastructure was not capable enough to keep up with the economic growth that was happening, plus having very large coastal populations. But what got buried in the sort of headlines when that paper came out was that the United States was number 20 out of 192 countries, and the EU collectively was number 18. And the reason for that is that while we have robust waste management infrastructure, we produce far more plastics per capita than any of those developing countries. And so and we have large coastal populations. And so even if we each lose just a little bit of plastic every day, like in that overflowing garbage can, it still adds up to a huge amount of waste. And a follow-on study that I led for the United States actually estimated that the leakage to the coastal environment from the United States might be as high as third if you take into account all the waste that we were collecting for recycling and shipping to Asia for processing, where it was likely not, the discards were not going into waste management infrastructure. I can't speak to the politics of those countries. I can tell you that in the World Bank report that followed that publication, China's numbers greatly improved, and they said they had invested in largely incineration, waste to energy plants to deal with plastic waste. And they also, of course, stopped importing our material collected for recycling. That door closed. And so that really caused a crisis in the world of how do we recycle all this? We were all sending it to Asia. So I do think that, work like this perhaps incited change. It might be because it was really just looked really bad for China to have risen to the top. And I hope that it's all for the good. And there's a tremendous amount of research coming out of China now. So I can't tell you how to solve the problem in these places, but I think that being willing to sort of share technologies and invest and figure out how the world can help places that need the help, and they're not just in Asia, deal with the problem. It's a global issue that we all have to tackle.
All right. Well, another round of applause for Kara for a nice presentation to kick off. And as Mario just mentioned before, we now have a coffee break of half an hour. So you can just pop outside and have a nice coffee, and then you can talk with all speakers if you want. I highly recommend that.
clever, but can't move the rocks.
For the gentleman, vodka martini, shaken, not stirred.
because I like to work in Antarctica because you only have one source. It's usually now you have two with the climate change, but usually you have the rates of transport but local usually is marked by PVDs, remember turtles, these are the only places, and I work in aromatic cameras because all the field bases have solar panels and windmills energy.
Yeah.
So they just turn on the gasoline generator and then you have electricity.
My friend is Argentinian, so she worked a lot in the Antarctica. She was telling me that it's getting contaminated to a degree that studying it as a like pristine environment doesn't
No,
make so much
when
sense
somebody
anymore. Yeah.
when people tell me that Antarctica is pristine, yes, probably it was pristine 40 years ago, but not now. Because I mean I performed something 2 years ago in the middle of Antarctica in a non-permanent field base. We were living in tents, like camping, and then switch on the generator using gasoil. So if it was pristine, now it's not. And this night I just sampled the air inside our campus, our area, our living area, 2 kilometers ago. I just look into the filters. Ones were gray and the others were white. So it was the local impact. Now we will have the results because of this pandemic. Because I sent all my samples to the Czech Republic. Yes, to relatives. Masaryk University. They are the best. They are the best. Because they invest a lot of money in that institution. And they have a support plan for Latin America. So I have a lot of friends there because I was doing my postdoc there. So if you need a contact, just tell me and we will give you a contact. Because also we have for students who they support them through the medicinal process. And it's not only just ECMS or ECE or ECMS. Also they have genomic facilities, they have programs. It's cheaper if you want to pay. I mean, everything is expensive, no? If you compare with Barcelona, where you live this year, or Canada. Canada was something that cost $1,000, and the rate was 500 euros. It's something. So it's easier for me and it's cheaper for me. And also I send my students because they will see a theoretical laboratory. Because I have a small PC here, which is not working anymore. It's like to have a most ECMS is like a most that you need to feed them every day. Usually, no, it's too much money. Usually I use my DC with the grad students. Give them right, great, and they learn how to work with the DC in the hands-on. Change the color to adjust the source, calibrate the signals. This is so nice for the students, but then for the kids, the students or the postdocs know how to do it. Okay?
Acá está el puntero.
Hablaban de la botella y yo me quería matar.
No, porque esta es blanca, ¿se puede reciclar?
Eh,
El problema son las de colorines.
sí. Bueno, este es el puntero. Y acá están los.
Ok. Sí, por la primera. Con la primera y listo.
Necesito un micrófono de mano.
No, tengo buena predicción de voz.
Es que es para el
Gracias. Vale.
So we come for a second. Ayúdame.
Permíteme un segundito.
Sí.
¿Está apagado? Presionas y se enciende.
¿Ahora está apagado?
Sí.
Ya, perfecto.
Sí, esperemos que entren todos porque primero yo voy a dar el pase, no sé si te van a presentar primero
Sí.
y después sale. Ya, perfecto. Entonces así nos aseguramos para que el streaming no se escuche nada antes. Perfecto. ¿Vale? Y tienes el punchline, estamos ok. Acá.
Synthetic pumps. They're soluble here so that they. Yeah, I have to look into that. I haven't looked into that. I mean, what's the chemical? There are many ones. And do we know how they interact with?
Me acuerdo cuando Fernando Mardones para el Congreso volvieron de veterinaria y tenía vuelta local de la cabeza. Venía moviendo porque no funcionaba la sala.
No. Tiene totales 45 minutos. 35 minutos.
Yo creo que voy a demorarme 30 minutos en la presentación porque es de 20 diapositivas.
Súper.
And they're fossil derived. They are polymers.
Sí. Okay, so we are back for the second half of the morning session, in which we're going to have two speakers. So the first speaker is going to be Cristóbal Galván Malagón. He's an environmental biologist from Universidad de Santiago de Compostela in Galicia, in Spain. A shocker for the speakers, because we had lunch yesterday, but they might not be aware that we just met yesterday as well. We didn't know each other in person before. We were communicating by emails during the stay that Cristóbal was doing in the US. And so we were trying to find a speaker that can explain a little bit of what's the context of plastic contamination, processes or mechanisms that involve contaminants and that are being done in our country. And so we decided to invite Cristobal to join us today. So he's going to be talking about micro-sized plastic particle accumulation in different trophic levels in Chile. Thank you, Cristobal, for coming. And yeah, please, the stage is all yours.
Thank you for the invitation. Okay, first of all, I will apologize because of my English. Okay, and to start my talk, I want to tell you something about my background. I come from environmental chemistry. I work on persistent organic pollutants, and plastics, curiously, are quite similar to persistent organic pollutants. So for me, was not very difficult to start this research line within my research team. How I start to study plastics? My first experience with plastic was when I was 26 years old. Now I have 40. And we found plastics in one research survey in the Arctic for my PhD. And I remember that when we took the troll with phytoplankton samples and all the zooplankton samples, I saw plastic particles. And I remember that I talked to my advisor, I say, oh, there is plastic there. And he said to me, yes, usually happens. But we didn't care about that. Usually happens. And it was in 2005. But remember, we were scientists working on pollution, and we didn't care about that because we were focused on persistent organic pollutants transport to the Arctic. So I finished my PhD, and then I came to Chile. I got a position in the Universidad Andrés Bello, in the facultad, in the extinct Ecology and Environment faculty, now Life Science faculty. And a student approached to me. I say, I want to work with microplastics. I say, okay, let's do it. But I didn't knew anything about that. So we start, and I will show you here some of our results there. Not all of them, because also we work with macroplastics at studying the impact at a population level. But I will show you the results of microplastics and what were our thoughts about the trophic transfer in the south of Chile. Okay, so we are working with not seals, otarids, which is like a sea lion. Okay, so first of all, probably you will remember this from the 2 talks in the morning. Okay, how plastic reaches the water. Okay, we understand that plastics, especially microplastics, have 2 kinds of origins. Okay, primary origin, which is microplastic just designed with this size and this shape and this color with a purpose. For example, scrubs, facial cleanings, etc. But our problem is that this secondary source, which is plastics that appear coming from macroplastics who have been weathered or degraded or partially broken due to the effect of light, the oxygen, mechanical abrasion, even also a biological broken. For example, a paper published by a Chilean colleague when he was doing her postdoc in Australia report that crabs are able to break these plastics, digest the plastic, and in the fecal pellets they appear like fibers. So as part of the biological or the daily life. But the thing is that macroplastics have one effect. Most of them is a level of, for the bigger animals, like entanglement, like gut blockage, for example, what we see in whales. But microplastics is another different history because first of all, they are not easily seen. You need to employ a logistics to study them and just pray to find them in your samples. You know, it's not easy to find them. There is, once microplastic reach the ocean, we have different processes. Microplastics are able to be part of this mechanical degradation for the waves, as mentioned before. We have biofouling. This plastics represents a plastisphere, and microbials start to grow. So it changes the density, and they don't float, so they could be in the mid-water column or in the deeper water column. And also, they could be eaten and could be part of the fecal pellets of copepods, for example, affecting also the carbon cycle in the water column. And then the other part that's very interesting to me is the weathering from ultraviolet radiation, which is quite important, especially on plastics. You go to the beach and take one plastic who was exposed during a lot of time, and you try to break it, it's more breakable than the new plastic. And this is the part that really interests me as a scientist. It's how they are able to enter the food web. How are they transported through the food web? And how are the things that tell us information about this accumulation? Because, as Carol said previously, there are a lot of reports of ingestion, but ingestion doesn't mean trophic transfer, I think. And one example is that we eat a fish, but we don't eat the stomachs of the fish, which is presumably where most of the plastic will be accumulated. But in the case of marine communities, they eat mostly the entire animals, excepting the sharks, because they bite. But usually they eat the entire fish. The prey is the entire fish. And this is like a study just telling us, this was Ungu et al. In 2021, just reporting what we do study in the water column. What is the animal group that we study usually? And most of them usually are the fish. And in fish, we are focused usually in the gastrointestinal tract and the stomach. Because we have a bias there. We think that every plastic should be there. And we have some experiments that I wouldn't show here, just reporting how they accumulate inside the animal and the effects on the digestive tract. And the others are the invertebrates. You see the water column, the two most important steps in the water column in the food web transfer are fish and invertebrates. Also phytoplankton, but plastics and phytoplankton would be a problem because they aggregate and they bond, but there are not too much reports on the phytoplankton aggregates in the open ocean, so it's very difficult. So we could assume that they are floating around, but there is no proof that this could occur in the natural ocean. In experiments, there were some experiments performed in the university in Germany, in Kiel, that they show that the aggregates are able to modify the phytoplankton performance and to affect the primary production, which is the base of the trophic web. So we have different trophic levels, and all of them are connected. There is no a linear connection because, for example, this big fish could consume here, 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 even here. It depends of the feeding strategy. But the problem is that we don't know the role of plastics here, and we know that this kind of animals could have plastics inside, and also this. So the food, or to feed on the lower trophic level, entails consumption of energy, entails uptake of carbon, and also classic pollutants, for example, as I mentioned, persistent organic pollutants like polychlorobiphenyls, polybromodiphenyl ethers, and pesticides, but also those emerging pollutants. Now, in environmental chemistry, plastics are believed to be emerging pollutants. Because what is an emerging pollutant? It's something that we don't care 20 years ago, but exists in the media. So we published a paper, all the researchers working in Chile on plastics, just look into what we have done in the last years. And it was a very good experience because we looked to each other What are you doing? What I'm doing? What can we do together? And similarly to the graphs that you see on plastic production, you see an increase in the number of articles. I think the numbers are not, but we are a small community. So when we look to what kind of plastics did we study, we were centered in the microplastics, especially in the last 5 years, because we learned during these years how to study microplastics. We learned, we got the technology, because, for example, Cesar told at the start that most of the studies are centered in the Northern Hemisphere. But our problem was the access to technology. I remember that 6 years ago, all the researchers of this network have access to one FTR. Because material scientists have FTR, but they were like, do you want to touch my FTR? Really? So we built our own libraries, because libraries were expensive, and say, okay, we don't want to buy a library, we will construct our library. And now, for example, in my research group, we have one micro FTR, which is working well. It has 2 months in the laboratory, but we are doing every day working on that, because we are starting to do a lot of things, especially with grad students who come to the laboratory. Just they want to work with microplastics. So here is the equipment. Let's do it. And the other thing is that we start to collaborate with people from the Northern Hemisphere, because we knew each other in the first Congress. For example, if you look to the papers that we publish here, we say, okay, plastics are blue, and they are round, and that's all. Okay, now we see plastics are made from PVC, or we have polypropylene, or we have PIT, and also we are able to study, and they are full of polybrominated compounds that are flame retardants. So there was a change. Also, I don't show usually this graph, but also the impact factor. We hate the impact factor, but it means something sometimes. The impact factor of the journals that we published increased, because we increased and we got better doing our job. When we look to what we reported in Chile, these were the main topics, which is the entanglement, the ingestion, epibionts, dispersal, contaminant and microbes of microplastics, sea surface abundance and distribution, shore abundance, and review and social survey. This is one of the most important in Chile. I don't know if you know Martin Thiel. Martin Thiel is one of the, I think, probably will know the number of Científicos de la Basura. This was an initiative performed by Martin Thiel, and just working with the schools and just to report the plastic, where it comes, not only the plastic, but also the debris that appears, where it comes, where it appears, what is the occurrence, can we do something, not just taking out from the beach, trying to make people to understand how it behaves in the environment, this debris. But ingestion, as you can see, because the entanglement was this part here. But as you can see, as I told you, we increased our knowledge and we start to report the ingestion, because we knew more about that and we knew better how to do that. Ingestion was reported in the whole coast of Chile, from different worlds. Most of them were on fish, of course, but also marine mammals. And also, we found this in Antarctic fish. It's in preparation. Sorry, I cannot show anything yet, because we are trying to understand our data. But we found, we take about 100 fish, like this size. They are called mactophytes. They are very common in the whole ocean. And we find plastics in 10 of them. But when we took samples from the coastal Antarctica, near the human settlements in Antarctica, the numbers increased a lot. I think we found plastic almost in every fish that we But you must think that it was the Antarctic island who has most presence of humans. We have King George Island is like the entryway of Antarctica for all the research stations and also for the tourism. So it makes sense where there is a lot of people hanging around, you will find a lot of plastic. So now I want to talk about where we found plastics, okay? Just to realize how this transfer could occur in a food web that we know very well, which is a food web from Waffle Island. Waffle Island is an island with a big colony of otarids. I think it's the biggest colony of otarids in the South Pacific Ocean, and it's the northern one. It's like here. And in the island there are only otarids and army people. There's something, 5 military, and then like 1,000 of otarids who live there, who raise their pups there. So we started to look for what they eat. And what they eat is king crab. Most of you will know because it's a very valuable economic source, especially for local communities. And what the group of Andrada Novando from the University of Magallanes found is that they found especially fibers. Most of them were passed through an FTR, and we see that most of them were made of nylon, and the materials that belongs to the long lines that the fishermen from the area use it. And that's what quite interesting. So we know that king crab from the deeper sediments, it's plastic, or at least contains plastic. What about the smaller guys? We don't have any report from Chile identifying copepods or similar to copepods, this kind of zooplankton, eating plastics. But we know that from other areas that usually appears. So this, I think, all of us know these images because we saw them probably on social networks. And not only they are able to eat them, but also they go with the fecal pellets and go to deeper waters, affecting the carbon cycle. But we know that these animals are able to eat and to retain them inside. Now we are going with fish, because fish eat copepods, or another fish. This was one experiment from the group of Martin Thiel, Nicolas Sori, a postdoc which is now in France, when they saw that fish are able to select their food because of their color. So you have copepods in the environment which are blue color, which are the smaller ones, okay? You see microplastics blue. You see that fish feeds preferentially on the blue. This is what they found. And we have like an inverse correlation. What you found in the fish, what you found in the copepods. That's quite curious. And this is what they look like. This is so curious, no? Because one person would think, but did the fish see blue like us? This is one of the things that was discussed, but it appears that from the physiology of oceanic fishes in general, we don't know about the specifics about that, oceanic fishes are able to distinguish from black, white, and some transition from blue to green, and they are not able to detect red or yellow, on the contrary to coastal fish. When they were able also to differentiate, this is the first work that we performed on microplastics in my group, and they were able to differentiate red from yellow, from blue from green, because of their feeding strategy. What we found here is that usually carnivores feed more on red and orange, herbivores feed more on brown, green, and something blue, and omnivores feed on everything that appears here. Because they are omnivores, they only need to grow and to eat and to eat and to eat to grow, because there is a part of their life in these tidal pools, which is areas that are very extreme for them because of the wave movements, and they got full of water, and then 2 days later they were run out of water, so it's quite difficult. And that was so important for us to detect this. And the problem with the fish is that most of the ingestion studies report the occurrence of microplastics in the digestive tract. We don't know if it is because of feeding or because of respiration, because they use the same mouth to take oxygen from water. We also exclude them, but this is not usually. I think when they feed, the digestive tract is blocked. Because we feed, we block the digestive tract, and then we do respiration. So it's a different way. But sometimes there are some reports of plastic fragments in the gills, similarly to what you say about mussels, because this is a filtration system. And when it appears, there might be involuntary. But we agree among the researchers that usually the consumption of plastic is because of confusion, because we don't know if it's plastic or no, the fish, but we need to eat, so in case of that, just buy them. This is what the animals do in these kind of things. Let's go to marine mammals. These were otarids from the study area, from Wafo Island, and we found these clearly macroplastics. Most of them were also composed by polypropylene, and some of them PVC and nylon, but in the area there is full of salmon culture. And nets is what they use more. And if you look to the colors, they are blue, because the nets are usually blue or green. The color could be changed because of weathering. We tested feces, because you are not able to take a knife and to open a, no. I think the ethics committee will kill us. So we use this indirect thing just to look at the feces, and of course, if there are plastics in the fecal pellets, probably will be plastics inside the animal. Okay, and there was one experiment conducted during this year, also in England, with in a zoo, when they gave plastic-contaminated fish to animals in captivity, just to know what enters the animal, what gets inside, and what goes outside. This was a very interesting work, because with these numbers, we could modelize how plastic behaves. You will understand later why. Also, we found this, not latitudinal gradient, I didn't put the latitudes, but this is Peru, this is Juan Fernandez, which is proximate to the South Pacific gyre. This is Pupuya Islet, which is very near from Santiago. Wafo Island, Chulec, and Punta Chayhuaco, which are in the Patagonia, and we see that islands usually, especially those islands that are inside the ocean, they are not very proximate to the coast, have a lot of plastics, because of oceanic transport will accumulate them. And also they have a deficient waste management, because it's an island. And Wafo Island was our area, but also we find levels of microfibers which were comparable to what we found in the island. So this is our animal. This is our case of study. And one of the things that for us was very important is not only to report if they are feeding plastics, or plastics have some occurrence inside the animal, is where did they come and where did they go? So this is not only to know the concentration inside the animal or the levels inside the animal, it's important to know how much they feed and how much they digest. So this is one of our main questions that we are working now. And for this, we use a similar approach that we use to persistent organic pollutants. Everything is about the ratios, induce ratio, uptake, digestion. We could argue that it could be some biotransformation, but not likely to be. Biotransformation is usually for more degradable compounds like plastics, like aromatic hydrocarbons, for example, that are more easy to degrade than a plastic. And there will be a growth dilution, but this is not the case. I will explain later, because plastic, in the best of the cases, have a half-life of days, from hours to days. In a copepod, probably the plastic will be some hours, is the transit time. And in these animals, the digestion takes 1 day, more or less. In whales, takes like weeks. And this is important because when you want to study the uptake and elimination processes in an animal, you have the loss for reproduction, but this is likely not to be, because plastics could be transferred, but it's not very, there is not much proof about that. But if you want to know the concentration, you know what they ate, and you know what they eliminate, then you're able to reproduce this through a food web. So you will know, okay, everything is fish, but we'll do a sketch. This is a marine mammal, this is a small fish or a crab, okay? So if you want to know what is here, you need to know this one. But you have some limitations, that is the uptake. You don't know the uptake. One of the things that is important is, okay, I have this Mike Toffee, who eats some copepods, for example. How much copepods eats per day? How much biomass it's per day? And inside this biomass, how much was the percentage of microplastics? So this is important to take into account. And this is what we are doing now. And the problem with this is the logistics. You must think that we travel 4 days to Waffle Island, or 5 days, my students, not me. And they spend like 1 or 2 months there working, but these sea lions doesn't stay there. They move around, especially the females during the growth season, during the rising season. And the other thing is that you need to collaborate with the fishers from the area. Just give me some fish that you take. You buy usually them. They don't give you give you for free. But now we are trying to figure out how to estimate this, because we saw one paper working with salmon, and they know, because salmon is a very important economic species, so they know exactly, not exactly, but with a good confidence what they ate, how many they eat, everything. They know everything about the salmon. So they were able to predict how many plastics will be inside the salmon, and how many plastic will be outside, and how many will be the uptake. So we are trying to replicate this with our limitations, and now we're planning the next year field survey, just to try to estimate, because you never will get the final number. You will get the estimates of that, and you will have a ratio of confidence, just to know and to figure out how plastics are transferred. But as far as I know, there is no a characterized food with this plastic transfer. And I know because of that, because there is no information, is because the logistics are very complicated, and also your number of samples should be very high, just to take confidence of what you're saying is not like a photograph. Just you need an album to prove something, not just a photo. So I think this is our main message. We want to working on that, on bioaccumulation. We are doing it now, and we are doing it in the end of the world, in the Patagonian region. So that's all, and thanks you for attending in the morning.
There's time for 2 questions if there's any questions in the audience. Otherwise, I'll start with one. There's one? Okay. Felipe, here you go.
Hi there, I'm Felipe, a student from Caesar Lab. And I would like to know about the state of the art in the impact of the plastics in mammals or birds in the marine environment. Because I read, I don't know the year of the paper, but I read that they were estimating, I don't know how to say this, estimando?
Yes.
Well, yeah, estimating like 1 million mammals and birds from the marine environment dying annually because of plastics. I don't know if that number is correct or if there is another estimation now. So yeah.
No, but
Thank you.
I think I probably heard about what you said because there was one study, for example, looking to birds from people from Australia and looking what are the deaths based on the occurrence of dead animals retrieving. But you must think that these studies are based on data reporting, and this is the same that Cara said previously about the production of waste for the countries. It depends on the data, but I think it's not important to take about numbers because you say, oh, there is 1 million birds dying or there is 100,000 animals dying. I think more than the exact numbers, we need to look at what are the process that is affecting that animals and what we can do to avoid this. For example, when people talk to me, okay, but plastic will be easier. You just take them, like the approach of Bojan Slat, for example. They take him and he will solutionate the problem. The problem is not to collect them in the open sea. The problem should be here in the land, no? And going to your question, for example, we are working in a report. Now I expect to submit the paper by the next month, working on the impact of on otarids, not only otarids, also in falsids, focas y leones marinos, around the world. And we work with the Van Seville model of the larva, because plastics could behave similarly to a larva, and also with the reports of ingestion and entanglement, and also with the fishing effort doing by each country. You know, there is reported that how many boats are going to fish somewhere. So you are able to estimate the impact of this one. And despite of the number, for example, in Hawaii, there are a lot of animals stranded with plastics around the neck. In Chile, we have 10 in 6 years, for example, in Guafo Island. So it's not about the numbers. It's more about to understand the process that are affecting that animals. Once you understand the process, you will be able to tackle the problem more correctly. I think.
A while ago, I was reading some reports that, because you were talking about also persistent organic pollutants, some pollutants which are present in the marine environment can accumulate in the fatty tissue, especially of animals of higher trophic levels, such as whales, porpoises, whatever. And we have previously heard that some of these compounds can sorb onto plastics. Is there any evidence that such compounds which sorb onto plastics can actually be transferred to animals?
There is a lot of evidence. I will show you. There is a global network of passive samplers of pollutants in water, and we use polyvinyl acetate to collect the pollutants because they equilibrate. I think now we are going to change to chemistry because we are talking about the partition coefficients between water and plastics. So you have one pollutant, let's say naphthalene, which is very volatile and is not very hydrophobic. It will be more soluble in water. But let's talk about pyrene or benzoapyrene, which is more hydrophobic and is not very soluble in water. So when we want to measure these compounds in the water without electricity and with a very simple logistics, because when I sample air in Antarctica or water in Antarctica, I think we talk about a lot about our logistics, but we go with very big equipments. We need a lot of electricity, but the passive samplers are made of plastic, most of them, because most of these compounds partition preferentially to this plastic to water. So we use them to measure and to give and to have an idea of how the amount of pollutants that are floating around. Also, there are some research groups that are doing this on animals. They put a plastic strip inside the skin of the animal, and they were able to predict the concentration of the animal, just measuring the strip, which is less invasive to kill the animal and to take a sample. And this is based on fugacity approach, which is a classical in physical chemistry and environmental chemistry. But yes, you have a lot. For example, one other example, we sampled plastics in the Corcovado Gulf, which is in the south of Chile, and we need to dilute the samples for PBDs up to 10 times because it was saturation, our GCMS, because probably they came from an installation who needs a lot of flame retardant, or the plastic has a lot of flame retardants. But we need to dilute because the equipment was, when you're seeing the signal, they go far, 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 and the equipment started to not working very well. So we dilute, for plastics, we dilute a lot of the samples. And it's in a semi-pristine area, which is the Patagonia.
I'll do just a very quick last question, and it will be maybe an opportunity for self-criticism as a scientific community. And the question will be, we are always described that we are a very long, narrow country, right? Which means in the end that we have a lot of coast. Yeah. So in your opinion, do you think that we're doing enough in terms of a scientific community to try to sample these very diverse areas all across the country to try to understand better what's happening with plastics and additives?
The thing with the plastics community in Chile, we have a network, which is called the SPLATCH, Scientific Plastic Alliance from Chile. We will have a meeting on Monday, online. And we are a small community of talking in PIs, in the principal investigator. We are 15. There are a lot of students starting now. But the thing is that, for example, when Cara talked about this meeting who was conducted in Uruguay and now will be in Paris this year for plastic, we don't know which is in charge in the Ministry of Environment about that. But even also, I have been working in pesticides and organic pollutants, and I don't know the people in charge of UNEP, because when I send an email to the contact that they don't answer the emails. But this is not the problem, because finally we got them. The problem is that when you are talking about policies in Chile, what are the main environmental problems that you have in Chile now? We have heavy industry pollution, you see Quintero. You have urban pollution. So, our invited people come in summer, but if you come here in winter, it's like London. There is a lot of air pollution in the city. We call this smog, because it's smoke plus fog, and you will know this when you breathe. And there is one of the other main problems. The other problems for the Ministry of Environment and Agriculture, the water. There is no water here. The wastewater treatment. So when you are going to the policymakers and say plastics, they say, okay, and they have a lot of problems. They need to solve, need to take care. But in the case of plastics, one of the things that was most important is because we have the first international law forbidding the single-use plastic. We were the first country in the world to ban the use, but it's banned in the law, but not in the practice. Just go outside the university and you will take single-use plastic easily. But the thing is that this policy comes from the people. I think this is most important. In the case of plastics and the plastic pollution, most of the policymakers are pushed by non-governmental organizations, by local communities that say, okay, we should stop this, and then it starts to go up in the policy. This is the exact case that happened in Chile. And because usually when you are going to top-down policies are more environmental impact services, other things that are more in the big area, in the big economy, you know. But this kind of laws, which comes from local communities, I think the first normative that appeared in Chile local was with a small, I don't know how to say, a small town. Say, we don't want to sell plastics anymore, and they forbid locally. And then this start to go to another areas, and now it's expanded to all of Chile. Now, for example, if you go to the supermarket, you are not able to take one plastic bag. If you go to Europe or the United States, plastic bags goes around everywhere. So this is the case here.
All right, well, thank you so much. Oh, I'll use this one. Thank you so much for the talk. And now we move on to... Okay. And so we now move on to the last talk for this morning, which will be perfect as a transition for what we're going to be discussing in the afternoon. So we've been talking about plastic pollution, the presence in different environments, but then we already know that there might be some potential alternatives for biological, at least degradation, potentially recycling of plastics, some of them, not all of them. And so for a transition, we will have Beatriz Diaz, who's a professor from the Catholic University of the School of Biological Sciences. We started collaborating 4 years ago, I think, something like that. And she's an expert in what will be metagenomics and collecting samples from different marine environments, especially Antarctic polluted environments, but also like every type of extreme environment that we have in our country, which we have many. And taking samples is trying to understand what's the ecology of those microbial systems. She got a PhD in the Universidad Autónoma Barcelona in Spain. So this is the second speaker that is actually working in Chile, but it's from Spain, which is quite exciting and quite a trip. But she's been around for a while. We've been doing a lot of collaborations, and one of the things that she's going to be discussing today is something that we work as a collaboration, but it was mainly driven by her extremely well-known background on metagenomics and omics in general and microbiology. So Beatriz is going to be talking about a global metagenomic survey of polyester hydrolases, and that's why I'm saying this is going to be a transition for the talks in the afternoon, especially in marine environments. So thank you so much for coming and for organizing this symposium.
Well. Hello, everyone. Thank you very much, Cesar, for two things. First, for organizing this, which is a lot of effort. And I think it's very good that we could have that here in the southern hemisphere, where we have been doing much less in the last years about this problem. So hopefully it will be coming more work from the southern hemisphere and from South America to put more effort in the coming years and in the coming future to increase the number of studies related to this big problem, global problem we have. And the second is that thank you, Cesar, to approach to me, even though our students, they approach to each other and then decide that let's do something together. And then involve my group and me in this complex, challenging, but very fascinating problem. So that's something that has been shown before, and I have that from before, huh? But it's something that seems like inspire many people, which is like seeing Earth from abroad. And that make my brain think that we have to treat problems in a global scale and seeing Earth from abroad. And then work together, join effort from different disciplines of science and other disciplines in life to try to solve such a kind of global problems. So talking about global, if there are some organisms in the planet that they are global in terms of dominate every single environment in Earth are microbes. And it's not because I work with them, it's because it's just the truth. They have been in the planet for so long time, much than we were here. And they have enough time to diverge and evolve, getting a lot of strategies and adaptations that make them useful for many things, like surviving under really harsh environmental conditions like high temperature, low temperature, pH, pressure, UV radiation, and many others, including pollutants and plastics, even though those are emerging pollutants, as they have been discussed so far. But there are microbes that they have been tested, and they can efficiently degrade many different things, like recalcitrant, mutagenic. No? Yeah, yeah, okay. So they have been there, and they have been efficient to degrade many different compounds, like xenobiotic compounds in general, like hydrocarbons, phenols, pesticides, etc. But then it's not weird to think that as plastics come from, or they are synthesized from hydrocarbons and petroleum, they will be also somehow used through microbes as a carbon source, mainly. So there are different organisms that have been already isolated, mainly from contaminated landfill sites, but also from other environments. And we have been working with them, not me myself, but the scientific community have been working with them for a while. But we know that they have to become one alternative, which is like more eco-friendly. That's everything else that Cara has been talking about, that is not really efficient. And then we end up in poor management and then accumulation in the environment. So this is something, and these are numbers that has been around here today already, so I'm not going to focus and repeat them again. But one of the environment that it's been talking today a lot is the ocean, and how we are accumulating these kind of problematic compounds, the plastics, in that specific environment. So basically everything that comes in terms of plastic to the environment are coming from mainland, and only a small percentage it's coming directly from something that is happening already in the sea. So as I say, microbes become like an eco-friendly alternative for all the standard things that we are trying to do to reduce plastic. Because they can use it, as I say, as a carbon source. So today I'm going to talk about some of the work that we have been doing with CESAR Group, and it's related to just one of the many different plastics that there are around in different environments, including the ocean, which is PET. So polyethylene terephthalate, that is always complicated to me, say that. So PET, from now on, can be degraded more or less slowly by microbes that has some enzymes called, because of this type of plastic, petases. So these are hydrolases that can degrade this PET to monomeric mono 2-hydroxyethyl terephthalate. And that is something that can keep going degrade and then use later on for being metabolized for microbes. So for many years, we isolate microbes and some enzymes from those microbes coming from thermophilic species. because PTSs work fine at high temperatures. And then we decide to look for them in these extremophiles that they grow at high temperature. But then we realized that maybe it would be very nice to have a species of microbes that they could degrade also at lower temperature. And then for that, we were looking for them. And in 2016, Josiva and collaborators managed to isolate one of mesophil bacteria, very well known, that is Idionella sakaiensis. And many work has been done with this bacteria since that. Because basically that one has all the pathways to, with all the enzymes that can degrade more or less complete the PET. So 2 years later than Josiva managed to isolate these bacteria, Austin and collaborators isolate the PTAs, and then they analyze the structural of these PTAs, and they found that they were very similar in structure to other PTAs coming from thermophilic bacteria. So it would work exactly the same, but at lower temperature. But what is the problem of isolating new bacteria with new potential PTAses that can be used for increasing degradation and different temperatures? Culturing. That is complicated. Isolate and culture in pure culture microbes is not easy. It seems like easy, but it's not. In general, it's very costly to have that. So basically, what is known is that from the environment, only 1% of microbes can be cultivated. And sometimes even less than this in some hard, in environments with really hard conditions. Even though if you manage to get them in culture, sometimes you cannot work with them because they are not nice with us. And they are not like open to share everything they do. So physiologically speaking, it's hard to work with them sometimes. And even though if you manage to do that, sometimes they need other microbes, the ones that they live together in the environment, to really show you everything that they can do. So that's complicated because sometimes we have them in pure culture, which means that they are alone. And we don't know what they do more than what we have been testing. So it means that we don't know what are the connections they need from others that they were living in the community. Even though we managed to do all this, sometimes we sequence the genomes and we think like, okay, let's get everything they do because we just can follow the genes and the pathways, enzymatic pathways, and then, okay, we know exactly what they can do and the potential capacities they have, but sometimes one genome doesn't really represent the whole species. So it means that you maybe can isolate one bacteria, you get the genome, but that doesn't mean that that one has all the potential of this species, not even the genus, family, order, and the phylum itself. So that happens, and we realize that culture in pure culture, bacteria or any other microbe is not easy, and sometimes it's basically impossible. So we have to develop new technologies to get all the information out of these guys. What we did is just culture-independent methodologies related like metaomics and, in principle, metagenomics. So here, with this technology, we managed to, in principle, access to the 100% of the genetic resources in one environment, and then everything that is in the microbial community. That has been done for many, many years already. And in the marine system has been used quite a lot. So what can benefit, what can be done using metaomics? Basically, it's an alternative for culturing. You can analyze complex microbial communities in many number of samples at the same time. It's better reproducible, and you can get information enough, maybe, to understand a little bit more about the context, and then manage to culture things that you were unable to culture before. And then you can also study microbes, enzymes, genes, as I'm going to show you in a minute, distribution, and you can make catalogs of genes, known and unknown ones, enzymes, and finally get genomes out of the metagenomes. And I'm going to show you how to do that. Then, with all that information, we will have access to genetics and metabolic potential, really, of everything that we didn't be able to culture. For many years, many people have been using metabolomics in a way that come from the cultures or enrichments, and then use them somehow to get the real activity on determined substrates, like PET, for instance. But what it has been less used is just using directly the environmental sample to, without any kind of culturing or disturbing the sample, do metabolomics, and then get all the potential that these microbes have for any kind of degradation or capacity. So microbiomes coming from all these samples, and in this case, environmental microbiomes can help us to understand the impacts and then try to help us with the solutions. So now I'm going to start with the real talk here, which is like talking about the marine plastisphere and the microbiome that is attached to that. The plastisphere is, by definition, a new ecosystem related to plastics and microbes living on those plastics. So for many years, we know now, and that has been said before during the day, that plastic is in the ocean, right? And more recently, there are some reviews that show how they really attach to the plastic and how the whole microbial consortia work together to live in these pieces of plastic. There are different examples of how we have been working using metabolomics in different ways to analyze the microbial communities living on plastic debris and how they develop, like in this very nice paper from Amaral and Seller and others in 2015, where they show how microbes develop in a microbial film on top of a plastic debris. So first, what happened is that diatoms, these photoautotrophic organisms, microorganisms, they get first, and they somehow get enough well the system for the rest to attach and then start growing on top of these plastics. And then after some weeks, the biofilm mature, and then many of them are more similar between them. When they compare different plastics from different environments, different marine environments, what they found is that basically we always find the same ones, the same microbes, in different abundances, but basically the same. So there is kind of a marker community that is more or less always attached to these kind of plastics. There is also nice study, the first one that was developed in the deep sea, microdebris, where these authors reveal that there are differences between the different kind of macrodebris. So not all communities are the same in each kind of plastic or in each kind of debris. And when we compare plastic with many other materials, plastic have their own marker communities. There was this other study in 2016 in the Ed DeLong laboratory, where they did the real first metagenomic using capacities, not only targeting or trying to understand who is there, but going a little bit farther and then see what they are doing or which are their capacities. And then what they just found basically is that they were metabolically active, and they have different genes or transcripts at that time working in these communities that they were attached to the plastic. But also, one very interesting thing is that they prove that they were completely different, not only in taxonomy, but also in the capacities they were using compared with the surrounding water. So it means that these communities are more or less specialized to work on plastic or to live on plastic. And that is very interesting. So as I showed you before, and others have shown before this review, that is fantastic, I must say. Again, here we see that the North Hemisphere has been the one that has been using also more metaomics, at least in terms of high throughput DNA sequencing. So that was in 2020. And then the main things that, or conclusions of this review was that some of them, I have mentioned them before, that microbial communities on these plastics debris, in pieces of plastic living or floating on the ocean, were different from other particles and less diverse in terms of type of species compared with other environments. That some of the groups that grow on these particular pieces of plastic are always found there. But none of these microorganisms were unique for this specific ecosystem. So there are organisms that they already live somewhere else, and then they attach, and they become or evolve in a way that they are very specific to be living under this specific compound. They also realized that there are some specific bacteria that they are very abundant in the ocean, and they are not there. So they like to live outside of the plastic. So there is here a very nice way to think that not all microbes like plastic, and some have the enough strategies for using them or attached to them or whatever. And that is something that we have to keep learning from that. So yeah, so it's a lot of things needed to do in the south, right? In the south and also in the polar region, because we don't see much until 2020 in the literature related to microbes and degradation of plastics in the literature. So it's something else that was missing there. So why we need to do more research on plastic degradation enzymes in microbial communities and their prevalence in the global ocean, and particularly in the polar regions? Well, I hope so far we have some kind of answers to that. But in, I think it was in 2018, there was a group of Danso and collaborators that they decide to use metagenomics, both in marine and terrestrial systems, to understand more, not on the taxonomy, not on the microbes living on plastic, but on the function itself and the distribution. So what they did, actually, is just use a methodology that was working at that time, and now we work even more, that is the Hayden-Markov models. These are conserved protein motifs that can be used instead of the whole homology of the sequence to target specific places in the sequence that they are very important for the protein itself, and then use it to target as a matter of consensus sequence, try to target the gene or the enzyme that we are of interest. In that case, they try to target PET enzymes. And they did alignments with everything, with all the sequences that were at that time on the databases that were able, or they were proved and validated as enzymes that have activity to degrade PET. There were only 9 or something like that. So they have to do a model with the sequences that we have available, including Idionella saccagensis that was described that same year. So they did that, and they found like different motifs that they were used for somehow target a specific site inside of the protein to get a consensus sequence that could be used as a query for looking at metagenomes. That's exactly what they did. And as you can see, again, in the north hemisphere, they use like different metagenomes and genomes from marine and terrestrial. And in some of them, they found a signal from these enzymes. They have more frequency in the marine than in the terrestrial. And there were differences between the taxonomical association of these particular enzymes related to microbes. So there were differences here, and that was important to have. So there is a paper in 2020, after the review, that is not so far, I understand, published. It's just a reprint that use the same target that Danso and collaborators to, in this time, use more metagenomes and try to see the prevalence of these PTAs on global ocean microbes. So what they found is more than 60 new PTAs from more than 40 samples or metagenomes that they come from the whole ocean, including the southern hemisphere, still lacking the polar regions. But they did a good job. They increased the number of metagenomes coming from the ocean, from north and south. And they found new things, okay? But why then, why then keep going with that work? So what is missing here? We know now that they are in the south, they are in the north, they are a quite number of new sequences coming from those enzymes. So why we don't keep calm with that and then keep working on those that we already have? But what is missing here? What is missing here is that, as I told you before at the beginning of my presentation, we have a lot of sequences coming from thermophiles. But what happened with other temperatures? And the ones that have been described in the south and in the north hemisphere are all coming from thermophiles or mesophiles, but not that much from organisms that they are psychrophiles. And then it's easy to think that maybe those enzymes coming from those organisms could have a bigger range of potential in terms of temperature. So that's why, and because we know that plastic is also in the poles. I mean, there are different publications, including that one in 2017, that prove that there is a lot of accumulation of plastic in the Arctic, which means that probably there will be organisms that are attached or around, and they will use and degrade this plastic. So all this bring us together with Cesar, and then at that time he was thinking about getting these sequences from the databases that were coming from organisms that they were already isolated or they were working at lower temperatures like those ones, like Morastella, coming from Antarctica and Oleospira. So they managed to get those sequences from the database and then work with them and prove that they can hydrolyze PET at lower temperatures than even Idionella. So that was the first thing that when they come to us in my group, say, okay, if these two can do that, there will be more sequences around in the Arctic or Antarctica that can help us to understand more about this deficiency of these petases at low temperature. I say, okay, let's look for them, because at that time I was working for many years already in Antarctica, and particularly in Chile Bay, which is like our coastal observatory. We have been there since 2013. And in this specific island, in the middle of the South Shetland Islands, we have been working every summer taking samples. So at that time, we have like a couple of three, I can't remember exactly, metagenomes from this place. And then we said like, okay, look on those metagenomes and see if we can mine sequences related to the previous ones that he found in the public databases. So that's what we did. And then we found them. We found some sequences that were like very similar to the Morse one, which is like the PTase enzyme coming from the Moraxella that was actually isolated by Cesar, and then prove and validate that it can degrade actually PT. And then we found similar ones related to the same family. As we use as query to mine sequences from these metagenomes, use this Moraxella, it's normal that we got like sequences more similar to that one, that they are around. And they are not exactly the same. They have a diverged sequence, which means that they could have like better or not efficiency than the one that he tried before. So we try to get more information about that, and then we thought like, okay, are those sequences really abundant? So are those PTAs really abundant in these samples? And what we did is just target and count them, how many we have on those samples, and they were not that much. But then we thought, okay, but it's just because there are not that many organisms into the family Moraxella that they have that capacity, or what? And then what we did is just like extract all the 16S ribosomal genes from those metagenomes, and then we quantify how many Moraxella species we have, how many members of the family we have. And then we compare them, and then we saw that the numbers were quite high. So what is going on here? We think that is going on is that not all members of this family, so not all species or genera from this family have the capacity to degrade this plastic, which means that, again, it seems like there are some species that really have potential strategy specializing to work on those ones, and not the whole family, not the whole genera, not the whole species have the capacity from one phylum or one family or one order to do that. So that is very interesting, why they select themselves to do that. So we keep going with that, and then we thought like, why don't we open our mind a little bit more, and then when we finish the first work, that Cesar is going to talk a little bit more about the chemical part and the protein perspective part, we move to trying to understand more about if we can mine even more sequences from, in this case, not only Antarctica, but also the Arctic regions, and compare with the rest of the ocean. That's what we did. And basically, we got all these samples coming from basically the same samples that the previous studies use, coming from temperate waters, but we also add a complete catalog coming from the Arctic Ocean, and some others new ones coming from the Antarctica, including new metagenomes that we at that time managed to have sequenced. So with all this, and at different depth, not only surface, but also samples on metagenomes that they were a little bit taken into the column water, we managed to have a huge catalog of metagenomes that we use to target PTases. PTases that were coming as a query from different type of sequences, the ones that were validated. We use basically the same that Danzo used before, but we include the ones that Cesar already had in his group. And then we make it a little bit broader, the Markov model, to be more specific for, or more open in this case, for looking for this specific low temperature, potential low temperature hydrolysis. So we did that, and then we target them on the predicted protein database that we obtain from all these metagenomes from all over the world now in terms of ocean. And then we managed to have a catalog of not only petaces, but also we did the same. We use this methodology also for other enzymes like that they can degrade completely the PET. So we did that, and that is something that is very new because we were not only working with petaces, that we got quite a lot of a number of new potential petaces from the whole set of the ocean, but also metases and then terephthalenic acid enzymes, related enzymes, which basically means that this part here is just like the microbe can metabolize this final product of the degradation of PET. So what we did is just like classify them using the same motifs that did Danso and Alan in their studies, and then we did a phylogeny just to understand more about which are the microbes that are attached or they have those petaces. And we did that. And then as you can see here, we have 4 clades. And this one is the most interesting, and I'm going to talk to you a little bit more about if I have time. These 4 is where all the reference sequences were before. So we have reference 4 sequences coming from other studies in these 4, but there is a bunch of other sequences that we have to take care later on of them because they are completely new and they have no other reference before that has been targeted for hydrolysis activity. So that's something that we have actually to think about. But let's focus here. And then in these 4 clade, what's interesting for us because it has all the main the main motifs, more complete and more complex motif, which have like all the sites in the sequence that they are important for the petaces. And then that's why we decided to keep working with this clade, what we did. And then what is important is that most of the sequences from petaces that have been proved and test and validate for activity are actually inside of this clade. So it's obvious that if we wanted to know more about new petaces with potential activity, we should go first for analyzing the ones that are in this clade. And I will go to that later on, but we have another question. Are those very abundant in polar regions? Because that was one of the main questions, right? And maybe some of them are just, or all of them are coming from Tara oceans, which means like these temperate samples or metagenomes. So maybe they are not actually that interesting for us because we are looking for enzymes that they can efficiently degrade that at lower temperatures. So we look for that. And then for that, we count them. I'm not going to enter how we did that because I don't have the time, but basically we normalize and we count them. And what we see is that from 59 polar and 43 temperate metagenomes, we found that for the different sequences of petaces, we have like basically Antarctica and Arctic have more. They have more frequently these kind of petaces compared with the rest of the ocean that is just here. So really, the polar region is one of the places in the world that we should go and look for to get new input about potential efficiency petaces. We also did the same not only with the petaces, but also with the other enzymes that are related to the complete degradation of the PET. And we found exactly the same. So actually, the whole thing that is going on there with the degradation, the PAT, have more potential in polar regions than in the rest of the oceans. Okay, we'll see what we do with that. So I don't know if I have the time, maybe just 1 minute, just saying that we wanted, now that we know that polar regions are important for looking for these enzymes, what about the taxonomy? What about the guys that have those pitases? Who are they? Are they important for the marine system? Are they specialists? I don't know. So what we thought is like, okay, let's get genomes from these same metagenomes and target those pitases, and then see how frequent those genomes have one, two, or the whole pathway for degradation of PAT. That's what we did, and that is the result. All these results are unpublished, so don't say anyone. But we managed to get like 120 max, which is these genomes coming from metagenomes that we can assemble. And then we found like in 4 of them, we only have pitases. In 15 of them, we have both pitases and metases. So we can go one step further on the degradation of the PAT. And then only the final product of the degradation, and they, since that they can maybe not degrade the whole PAT, but they can use the substrate, the product of that, to metabolize it. And then only one has the 3 of them. So means that this one is a champion. This is the one that we should look forward and see who is that one and why that one has that potential with everything there, or maybe it's just like something weird that happened with the assembly. I don't know. We have to go through that. But these are the numbers, and this is what we have in our hands right now. So I'm not going to explain that, but basically this is the most important message here, that we have different genomes that we know that they have potential, different kind of potential, which means that there must be a consortia here, that they're working together synergistically for finally degrade somehow these kind of enzymes. We'll see how it's all that. So coming back again to, and that one I finished, I promise, to this clade 4, where all these sequences from the reference with proof capacity of hydrolase PET were, and then we realized that they have 2 clades. And in one clade, we have all the sequences that are thermophilic. And in the second clade, we have most of the mesophilic ones. So what should we do? Just choose both of them. Some sequences coming from the thermophilic, because those are the ones that always degrade. And then maybe we have the chance to get something new and very efficient, even though maybe they are coming from psychophilic microbes, and also from the lower temperature clade. And then we select 5 and 5, just to be equal, okay? So that's what we did. And I'm not going to tell you about this, because I know that Cesar in the afternoon is going to tell you about purification. Once you have these sequences, then a new world opens in front of your eyes, and means that you can use these sequences to purify the enzyme somehow, and then try to see if those enzymes really actively can degrade your substrates, in this case, plastics. And the only thing I'm going to show you is that it's the work is promising, because from these 10, at least 7, we got confirmation of at least some polyester activity. Now we are working on that, and Cesar is going to show you in the afternoon how they are doing all this to get the final closed cycle of this study. So yeah, so I'm not going to go to the conclusions, because I think we have been talking about and discussing about that enough. Well, yeah, I wanted to show you this paper that just showed up like, I think 1 year ago, more or less. And this is a way of doing everything I'm telling you in a more fancy and probably faster way. They managed to make a plastic database, they call, for microbes and proteins linked to plastic biodegradation. And you can find a lot of information there, and you can do things inside of this platform too, which is very interesting for people that maybe is not that used or is not used to work with metaomics. So you can put all the genomes here that you have, or use metagenomes that have been using for plastic biodegradation related things, and then compare them together and get some data out of this that can help you to understand a little bit which kind of questions I have to do to myself to keep going with research. So yeah, I think it's good for everyone just to have something, some kind of initiatives. And just finish my talk, just acknowledgements to funding, and definitely to Cesar again, thank you very much for involving me into such an amazing and complicated work that was done basically by Geronimo. I have to learn and study a lot just to tell you about all these things, because he's the one who did the job, okay? He's the one that talked with someone here and then discuss, should we do that? And then put together Cesar and I to discuss and put the money and put a little bit of our brains just to think how we should do that. But actually they did the work. Arantxa did a lot too, and Paula, and all of them did a lot of work just to get those results that hopefully they will be published soon, and we will be the first. So thank you very much to all of you for your attention.
All right, so we have time for one burning question if you have anything.
You can talk to me later on. I will
Yeah, otherwise you can talk later on. But if you do have one, just raise your hand. Otherwise, you just go for lunch. You have one, okay. You have to be the guy with the question.
Thank you. Well, my name is Gabriel. I have a question. Have you looked at using single cell sequencing for look for these kind of enzymes, or has anyone done that?
Fantastic question. I actually yesterday I was thinking why we don't do that. I have seen none in the literature, but I'm sure people is more into that right now, because it's the next step, basically. So now that we know about the sequences, now that we know that we can mine sequence directly from metagenomes and get genomes out of those metagenomes, it's obvious that the next step is just like, okay, try to get them isolate for actually asking questions in the lab too. And not only for genomes, but also for asking questions, because when you do like cytometry, for instance, to separate bacteria, you can do both. You can separate them and then grow in cultures if you manage to, of course, but you can at least get the genomes and then ask the same questions that I'm asking to my build artificial genomes coming from the metagenomes. So yes, I think there is a nice question, and I'm sure there is a lot of us thinking about doing that, but I haven't seen anything related to plastic degradation related to that methodology so far.
Thank you.
You're welcome.
Great. Okay, that's the closing of the morning session. So thank you again, Fea, for amazing talk. So just yeah, I'll switch here just for a few. No, it's not working. I'm gonna use this one. Just for a few announcements. So the room's gonna be closed until 2 p.m. So you better take your stuff out. We're gonna take a break for just free time for lunch. So we are back at 2 p.m. I'll just prime you into the things that we're gonna be discussing. So we'll be discussing about enzymes and degradation, and then microbial consortia degrading plastics, particularly PET. We have an amazing opening lecture by Wolfgang, which I'm looking forward to actually watch. And then it will be me with the Antarctic enzymes, and then lastly will be Bashak with also an amazing talk. Mine's gonna be fine. Theirs is gonna be wonderful. So looking forward for your presence here at 2 p.m. And then remember that we're going to have an ending social mixer after 5 p.m. So you'll stay all day here with us. Thank you so much. See you around.
¿te tinca ir también a eso? Entonces, terminándolo de la China, ¿qué te dijo la muchacha? No, cerramos entonces, igual que no estáis hablando.
There's definitely no matching to
¿cierto? Mientras no mate